1,33%, Banca Mic or yeah, okay. Can you hear me from everywhere else in the world now, or it's still not working?
Yes, much better. Thank you very much.
Okay, a lot of emotions this morning already. More emotions during the day, I can assure you. Yeah. Okay, can I have the double thing? The extended view, I mean, yeah. Okay, I think it's good because I just changed from here and I see the notes from here, so it's good. Okay. Can you do it back? Yeah, okay, okay, good. Okay.
So then, may I briefly interrupt and introduce you, Ariana, with all this hassles going on. Yeah, Simon, hi, you can hear us now. Good to have you on my back, virtually at least.
Yeah, I'm very sorry.
I'm gonna start with the session now, or maybe briefly introduce Simon also to the virtual audience. So Simon Eickhoff is full professor for systems neuroscience at in Düsseldorf, Heinrich-Heine University, and directing the Institute of Neuroscience and Medicine at the Research Center in Jülich. And his work focuses on multimodal image analysis and machine learning applied to brain mapping data and to disentangle inter-individual differences and apply to clinical questions. And we both host now the first session, so thank you very much for inviting me to my basically hometown, basically here, to moderate these interesting sessions and to find some common sense on this very tricky but also interesting and helpful topic for clinical and basic research.
Yeah, and I'm sorry that
let me introduce now one of the spearheads of this symposium, organizing it, Ariana. Ariana Sala is a postdoc research fellow at the Coma Science Group at University of Liège in Belgium. And after her PhD in molecular medicine, where she investigated molecular biomarkers in Alzheimer's disease, she joined the Coma Science Group to study now brain glucose metabolism and metabolic networks in disorders of consciousness. And yeah, she has been a visiting fellow here to Igor, and there's the connection, I guess. So she's co-founder of the Molecular Connectivity Working Group. And yeah, we're interesting to hear now your summary about the history of macroscale brain connectivity.
Okay, so thanks everyone for being here. Thanks a lot for the invitation. Actually, this is a very difficult talk for me because I'm speaking about the history of macroscale brain connectivity, so a bit all modalities, just before all the experts in these modalities are going to give their talk. So no anxiety at all. The history, let's see if this works. Yes. So if you look on PubMed for brain and connectivity, you will get thousands of results. You can see here that we start to speak about connectivity actually in the 19th century. So we have 140 years of history of brain connectivity that I have to cover in these 30 minutes. Very difficult. You see here, right now in 2024, it is expected that we will have around 10,000 papers published this year about brain and connectivity. So how does it all start? So it starts again in the 19th century. And we here at the beginning of history of connectivity, we are not at the macro scale level, we are at the micro scale level. So we start to speak about connectivity in the moment that we understand, thanks to Ramon y Cajal, that neurons are individual cells and they are connected to, with each other by means of dendrites and axons. So we start to speak about connectivity as something that we can really see, identify, it's something structural, and it's kind of self-evident. To speak about macro-scale connectivity, we have to wait a lot of years, around 70 years. So it is only in the 50s that we have what we could consider as macro-scale connectivity studies. So now we are very much used to see these kind of diagrams. So we have nodes, which normally represents, let's say, brain regions or areas that are defined based on some arbitrary, more or less, parcellation. And we have links or edges that represent link or connections between regions. And here we are at a very abstract level, right? This connection can be anything, can be structural connection, can be functional connections. So we have the same representation to speak about actually pretty different things. That's why I would like to change the title of my talk to History of Macroscale Brain Connectivities. And I will make this more clear by showing you how the different types of connectivities look like. So these you can see different fingerprints or, let's say, connectomes of connectivity computed using different neurophysiological tools. So you have the structural connectivity based on MRI, everything right, okay. We have the functional connectivity based on EEG, MEG, FNIRS, fMRI. And, sorry. Oh, it's not working. No. Yeah, and in the past, also studies based on PET perfusion and metabolism were considered as functional connectivity. And then we have a series of tracers that are made available by PET that allows us to assess more in general molecular connectivity. And you can see here, these are all data that are real data that were collected with a lot of effort from the people here below. And you can see how you can appreciate how different, of course, it's not the same subjects, but you can appreciate how different all the different types of connectivity are. And this is, I would say, not really surprising, right? Because here, it doesn't work. I need to use, may I? Yeah, okay, thanks. We are speaking about very different biological targets for different neurophysiological tools.
Online. So please
we
share
have
with us.
Some tools, usually the ones that are considered as measuring functional connectivity, like fMRI or FNIRS or perfusion PET, that measure something happening at the level of the blood vessels. Then we have some tools like MEG or EEG, and also structural MRI that are mainly measuring something at the postsynaptic level. We have, diffusion MRI that measures something at the level of the white matter. And then we have a series of molecular targets from the point of view of neurotransmission at postsynaptic and presynaptic level that again measure something completely different. And finally, we have PET allows also to measure proteinopathies, so pathological accumulation. And this is also something that has been studied in terms of networks and connectivity, as Igor will explain better later. Another important point to make, and I would like just to go through all these things, because the history of brain connectivities is based on very different modalities. And I think based on the modality we have in front, the definition, the way that things were described changed quite a lot. So we are speaking of different modalities with very different spatial scales. So here you can see how, so the highest resolution we have at the level of structural MRI, and you can see how going on at the level of functional modalities, the resolution progressively decreases. So what here looks like quite a quite detailed representation, here is just one value. And at the level, going a bit more at the biological level, you can see how when we speak about connectivity at macro scale, we are speaking about like average of either thousands to millions of neurons. So we are very, very far away from those initial, microscale neuron level connectivity from which we started from. The temporal scales of the modalities are also. Okay, so I will stop. Okay. So I have to start from the beginning again. No, it was enough. Okay. We also, as I was saying, speaking about modalities with very different temporal scales. So if we consider, I consider here as reference the time scale of fMRI, which is around 2.5 seconds. It takes around 2.5 seconds to get an image and the whole brain. And this is around 8 times faster compared to fNIRS, 600 times, sorry, slower than fNIRS, is 600 times slower than EEG and 1,000 times slower than MEG. And it's around 2,500 times slower than time it takes for an action potential to be transmitted. Compared to fMRI, we have PET, which is actually a little bit a slower technique. So in the best case, PET, functional PET is lower around 6 times compared to fMRI and around 700 times in the worst case. So again, here we're speaking again of averages of thousands to millions of neural events. Okay. I will start finally, we can start talking about the history of connectivity. And I would like to start with a question. So here you have all the different types of connectivity that, as far as I know, have been computed. And I would like to ask a question to the audience, and also on Zoom, you can reply on the chat. I would like to ask you, based on which type of data was macro-scale functional connectivity defined for the first time? Is it EEG or MEG, fMRI, PET, or something else? So I will go by raising your hands. So who says EEG and MEG? Okay. Who says fMRI? Not many, strange. And who says PET? Okay, so actually EEG is going quite strong in this audience. Interesting. Usually it's fMRI that tends to win the poll. But okay, let's see if you're right. So the first study that we can consider of macro-scale brain connectivity is a EEG study. In this study, we don't speak yet about connectivity, but we speak about cross-correlations. So this study really starts from the point of view of, let's say, an engineering point of view. It speaks about communication theory and just using communication theory to describe what's happening in the brain. What is done is that in this study, what they do is that they cross-correlate, let's say, the time series of EEG measured in different at different locations in the brain, or let's say at different electrodes. So we are at scalp level, and they see how these frequencies correlate with these time series correlate with each other, assuming different delays. And you see here at the bottom how in this case the highest correlation is obtained when we consider between two regions. Here it was left and right temporal, is around 50 milliseconds of delay. Even if they start from this mathematical perspective, they actually try to give a neurophysiological interpretation. And we say if we see this kind of delay, this cannot be by chance, and we can think it is due to tract conduction and synaptic delay. So we speak about EEG, but there must be some anatomical connection between these areas. There is some synaptic delays, and these areas are essentially communicating. Now, for 20 years, not much happens. So connectivity remains something that only EEG people do. They start to speak not much about cross-correlation, but more about coherence, which is essentially it's something like cross-correlation, but for specific frequencies. But in the 70s, we have actually the first scintigraphy regional relations study. So this is a technique, it's a molecular imaging technique used mainly in the clinics. It's a much lower resolution compared to PET, that still can be used to measure, for example, perfusion. And in this study in 77, they try to they describe just how blood flow appears in resting normal subjects. They don't really give any justification for doing this, but at some point in the method, they say that they compute interhemispheric regional relations based on product momentum correlations. And this is done through multiple measurements. So we have 22 subjects, but doing multiple measurements, doing different language tasks. And they compute the correlations across regions based on these kind of time and subject series. how do we interpret this? For them, this correlation is still related to anatomical coupling, so there must be an anatomical path between these regions. And why these regions are connected? Well, probably they are kind of doing some kind of coordination for bilateral information processing. So there is a sort of functional interpretation of why the regions are connected because there is an anatomical path between them, and they are connected because they need to do something together. So they speak about functional covariation or coupling, which is not so far, I would say, from what terms that we use still now. After that, we have the first studies of FTG PET, so we are measuring glucose metabolism, which is an indirect index of excitatory neurotransmission, I would say. And here we speak about interregional correlation. We have two studies in the 84, published almost at the same time. And here we speak not about, again, connectivity, we speak about metabolic relationships for selected brain regions. And you see here in the diagram on the right what kind, the regions they select. So it's not whole brain, they select some regions and they compute a correlation matrix. And this is done with metabolism at rest. So there is no task, it's just one measurement per subject. We don't look at the correlation in time series, we look at correlation across subjects with the idea that, let's say, we can say something about brain function by looking for, covariance, if regions can change together in time, but can change together also across subjects, and that's an index of some kind of association. They give a reason for doing so. So they say it's obvious that regions don't function in isolation, so we should study, their interconnections. And when metabolic changes occur in one region, we should expect changes to occur also in adjacent and distant regions, depending on the function, once again, what these regions need to do, and their innervation, which I intend, again, as an anatomical connection. So they do this, they find some interesting correlations, they find correlations among homologous areas, and how do they interpret it? In two ways. So one way is, again, the functional way, so they say these regions are metabolically related because the areas might be essentially working together in some tasks, like here, like visual processing, control of eye movements, or whatever. And they also give a second explanation saying that, again, these regions that we find strong correlations across hemispheres, this must be due to the presence of callosal connections. So these regions have yoked activities. So we don't know why it is yoked, but so the presence of an anatomical connection allows for a certain influence between the two regions. The next paper is a bit more detailed. So again, in the same year, we also have the paper by Horwitz. It doesn't speak about relations, it speaks about intercorrelations. So it goes a little bit at a more, let's say, statistical level. And it gives this definition that you will find quite often cited in FTG PET studies of connectivity in the years, saying that regions whose metabolism is significantly correlated are functionally associated, with the strength of the association being proportional to the magnitude of the correlation. So there is really this idea of regions being coupled, functionally coupled, and functional in the sense that they are coupled to do, again, something together, to make a certain task possible. And this happens also in the resting state. So how can we explain function in the resting state? Because there was this idea that the brain at rest is not doing much. And they say, well, at resting state, if we observe this relationship, this functional association is because the regions are doing something to maintain general consciousness. Again, how do they interpret all of this? Actually, the literature, it's quite a bit all the same. It's 20 years where people keep saying, really, yeah, we see this functional association because we have some anatomical connections. And also, perhaps, they give a bit more inferential explanation that the regions are coupled at the metabolic level because they are doing something together, maybe doing some kind of low-level monitoring of one another's activity. They also provide a third explanation, which is quite interesting. They say that maybe these regions are both activated in a similar way by the reticular activating system. And all of these options would lead to the observed interaction. Okay, so it's like one interaction, but kind of many different interpretations, more anatomical, more functional, and more, let's say, related to the arousal system of the brain. After this, we have finally the answer to the previous question. So when was functional connectivity defined? And before telling you how it was defined based on which kind of study, I will actually go a little bit backwards to the first occurrence of functional connectivity in the scientific literature. So the first occurrence in, let's say, macro scale level is in an EEG study, where they speak about coherence, and they say coherence is in 1990, saying that coherence could be viewed as an index of functional connectivity between different brain regions. And that sounded to me very weird because functional connectivity was not defined yet. It was defined in 93. So I guess, okay, so here they are referring either to an intuitive concept or someone else must have defined functional connectivity in another field, in another context. And if you go in the 60s, you find a whole paragraph about functional connectivity, speaking about the neuropil, which is essentially the equivalent of the brain across a wide range of animal species. And they speak about functional connectivity as a contrapose to structural connectivity. And they put this nice picture of functional connectivity, which is a spike train. So these are measurements of action potential at the at an individual neuronal level. And again, in the 70s, we find again people speaking about functional connections, considered in terms of temporal correlations that they induce in the respective spike trains of neurons. So actually, functional connectivity was first defined at the level of spike trains. And how do we interpret these correlations in spike trains? There is actually different hypotheses of why this happens. One option is that neurons might be connected synaptically, and you have here different possibly wiring diagrams. They also say maybe these neurons receive common input from presynaptic sources, or maybe it's not really related to, let's say, neural communication, but it's about a common dependence on some factors like blood, CO2, temperature, and blood pressure. Or also maybe some, let's say, top-down effects where there is a modulation of the excitability of the single neuron from the general population it belongs to. So this is a situation about functional connectivity between 1993. So it's about temporal correlation in spike trains. So we are at a microscale level. In 93, Freestone arrives using a PET study, perfusion PET study, and he kind of translates functional connectivity at a macro scale level, saying that functional connectivity represents the observed temporal correlation between two electro-neurophysiological measurements from different parts of the brain. And he is quite clear that functional connectivity for him can only mean something that is analyzed at a temporal level. So you cannot do this if you look at co-occurrences across subjects, for example. But this makes sense because, again, he really comes from this definition about spike trains, where functional connectivity was defined as temporal correlation across spike trains. So how do he connects the functional connectivity of spike trains to the functional connectivity of PET? He does it through EEG. So from the functional connectivity in spike trains, he jumps to define coherence as let's say a type of functional connectivity at a microscale level based on EEG, and then he does the last jump from EEG to PET, saying, okay, PET is also at a macroscale level like EEG, and we are just looking at some lower frequency components of the coherence, because of course perfusion is much slower than what we can measure with EEG. How is this interpreted? Again, there is a reference to anatomical pathways. There is also a reference to the possibility of some jointly pooled activity and or contribution from direct connection. So this is, again, these three interpretations are again present at the macro scale level, very similar to what was defined at the micro scale level. Okay. then I will go a bit faster because I have only 6 minutes, I see. We have the first functional MRI studies, where, again, we speak about connectivity. So the concept of connectivity is now taken and applied to different contexts. And here connectivity is defined, again, as the temporal response pattern during dynamic data acquisition. So here we are at task level. So at task level, we observe how time series change in time across different parts of the brain. And this is interpreted in a pretty, let's say, basic way, as we have a functional connectivity indicating that there is coherent activation across distant site. And here you have a map of functional connectivity. The next study in the same year is a REST study, task and rest study, where Biswal observes that the pattern of activation that you see during a finger tapping task is similar to what you observe as at rest if you compute functional connectivity as rest. And he says what we observe here is not about another task. It's not like the subject is doing some motor imagery task. It's not imagining to move his fingers. It's just how it is. So this correlation of low frequency fluctuations at rest is a manifestation of functional connectivity in the brain. What happens next? We also have the first structural MRI study of interregional correlation. Here it's not functional connectivity really. We are speaking about correlating gray matter volumes. Okay, so here this has been discussed later on as a sort of structural connectivity, but in the first paper they really discuss these in terms of association. And it's a little bit vague because they speak about patterns of association between regional volumes and of dissociation, with a bit the idea that there is a link between structure and function, and this correlation in gray matter volumes might be representative of something that happened, for example, during development. So that there is some trophic factors that are affecting the way the amount of gray matter volume develops during life. So it's a bit, let's say, it's not clear, like what it means right now. It's a bit about the idea that this association measure reflects what happened in life up until now. After that, we have an important milestone. So it's not a new type of connectivity, but we start to expand the concepts, the terminology that we use to describe connectivity and, let's say, this multivariate brain pattern. So we speak about the default mode network, which actually is defined at the beginning not as default mode network. We speak about networks of regions, and it's defined based on meta-analysis of perfusion PET studies that defining the regions where we observe consistent decreases during different tasks. So it's a sort of a negative network that will be defined as the default mode network in around 2000, but here we have already first concept during the end of the 90s, again based on PET perfusion studies. After that, we have the first MEG coherence study. Here, I would say, this is still functional connectivity, right? But actually the authors, at least in this study, are very careful to never speak about functional connectivity. They really speak about coherence and neural interaction. And actually they say that, if you want to measure coherence between brain regions, you cannot do it with functional MRI because it's too slow. You need an adequate temporal resolution to measure oscillatory activity. And so we can address the cortico-cortical interactions, which is a bit a vague term, with MEG to study the coupling within distributed cortical networks. So again, a concept a bit similar to what we want to study with functional connectivity. In another study, we have again a similar concept expressed. They speak again about coherence, but the point for them is to study functionally related areas. That they think are related because, again, there is an anatomical connection and there is a coupling between regions. And this can be seen not as something not so distant, actually, from functional connectivity from fMRI if we consider the low frequencies. we also have the first structural connectivity study. Just for completeness, I would like just to say that actually structural connectivity is introduced at macro scale level, speaking again about coherence and EEG, with the idea that people tended to interpret their EEG results as reflecting some sort of underlying structural connectivity. And again, they speak later on in the 90s about structural connectivity as the anatomical substrate of coherence. But again, then it is in 2001 that we have structural connectivity defined based on diffusion MRI as nerve fiber bundles in the white matter. And I think this is a very straightforward definition. Is the white matter bundles is something you can point at, you can see it. It's not particularly, and it's not as complicated as functional connectivity. Not last, but almost last milestone in the history of connectivity is then a description of resting state networks. And this happens not thanks to PET, but thanks to functional MRI, where we start speaking about functionally related regions, again, establishing a core association between what we observe at task as patterns of co-activation and at rest. Not differently from what Biswell did, just applied to different networks, the auditory network, motor network, language, and visual. And again, assuming that these areas are functionally related, functional in the sense that these areas subserve the same function, cognitive or sensory or whatever. Another study, very similar, is again about resting state networks, where they get defined really as networks. So before they spoke about related regions, here we start speaking about resting networks, again associated to interpreted based on their functional relevance, based on similarity in tasks. The last milestone is the definition of the connectome, which was defined as the connectome matrix of the brain. So it was defined at the beginning really as something related to anatomical connections, as the elements that are at the basis and put constraints on brain dynamics. And here you can see a first description, one of the first description of the brain connectome by Patrick Hagman, who is here, where it is defined as a comprehensive map of structural connections that is needed to understand functional connectivity patterns. Last thing that happens, and then the ancient history, it's done, is the first FNIRS study that actually appears after all of these has happened. Of course, it's a recent technological advancement. Here, let's say I would say there's not much to say. They speak about correlation maps, they speak about consistent cortical interactions, and they discuss what's the better way to measure functional connectivity. And here, I think it's interesting, and I'm done, because here they really speak about functional connectivity at a macro scale level. So we kind of have forgotten the spike trains, the micro scale level. Here we have one of the first examples when, using FNIRS, we try functional connectivity as we know it from fMRI, right? So if what we have is similar to what we observe with fMRI, then, well, it means that our measure means something. Okay, sorry, this was, I didn't cancel annotation, but so in summary, this is a bit of the overview of the history of macro-scale connectivity. So you have really every modality contributed to it one way or the others. Take-home messages, and then I will leave you. The history of macro-scale connectivity, again, developed around different neurophysiological tools. The milestones in particular were really driven by studies of perfusion PET and more recently of functional PET and functional MRI and then infusion MRI. And the concept of some brain connectivity has evolved through time. I think structural connectivity as white matter tracts is a very straightforward concept, but for functional connectivity is a bit more complicated because people spoke about cross correlations, interactions, so meaning something related to neural communication, but also not, spike trains. So it's a bit more elusive concept, which I think, as we will try to do, is very important to define very precisely, because the way we define it then defines how we can interpret it. So we must be very, very clear of what we mean when we use this kind of metrics of concepts. So that's it. Sorry if I went a bit over time, and thank you very much.
Wow, Ariella, is it working? Yeah. Wow, thanks Ariana for this beautiful overview. I mean, this was a massive endeavor. Yeah, I know. I'll come to that. That there are no questions for the moment, but thanks for still sticking in time with having two like restarts of the virtual session. I got some feedback that it was working now. So yeah, I hope that a lot of people online could join and enjoy that talk. Simon, you're also back. Hi, good to see you again. Hello. Sorry, we have no time for questions at the current stage, but we'll hear two more talks now where we can ask a few questions afterwards before we go into the first break. And as we had this delay in the beginning, so I'd suggest we just continue with the next talk. So the next talk, as we heard, beautifully summarized now by Ariana. So it all started a bit with neurophysiology, like spiking activity, electrophysiology. So I think it's only natural to continue now with some electrophysiological data. And the next speaker will be Martin Wink. He's a professor at neurophysics at the Donders in Nijmegen and a research group leader in Frankfurt in Germany at the Ernst Ringmann Institute at the MPI. And his work focuses on understanding the principles of neural communication and coding in the cerebral cortex. And he uses both causal approaches, very interesting to hear about that, like optogenetics, electrophysiology from mice, scaling it up to humans and computational modeling. Martin, the floor is yours.
Should I use this? Do you hear me in the back? Yeah?
Please use it for hearing impaired people.
I should use it, yeah? Then it's going to be loud. That's fine. So how do I... This is the... Sorry. Huh? Gotta use the mouse here? No, I think we have to put it full screen or something. No, that one doesn't work. All right. Okay. Good. Okay. Good morning, everybody. I'm Martin Vink, and I would like to start by thanking all the organizers here, Igor and all the others. This is quite a new field for me, molecular connectivity. When Igor invited me, I thought, okay, what is it? But he said, we want to learn a bit about what you are doing in EEG imaging. And I think Igor expects that things are very clear there, but they're actually not. So, I mean, And so our lab works a lot about on bridging different skills. So we go from spikes, memory potential, all the way up to EEG, MEG. And there's nothing harder actually than bridging these skills. And today we're actually talking about many different modalities, which makes it even harder, right? So our approach in the lab is to take a very bottom-up approach. So we start from large-scale models, theoretical models where we know the ground truth, or we also perform causal perturbations, for example, in animals using optogenetics, so that we can really study what functional connectivity measures actually mean, right? And that's what I'm mostly going to talk about today. So I'll start with a brief introduction about EEG, MEG measures, and give you some overview of the standard connectivity measures. And then I'll go really to the heart of it, so how to interpret measures like coherence, okay? And can we actually infer effective connectivity from functional connectivity? And that will lead me to motivate also the application of nonlinear measures to study brain function. So I think most of you are familiar with that, but I'll just repeat it. So the main electric signals in the brain, as very nicely introduced by the previous speakers, are spikes, memory potentials, so the internal voltages inside the neurons, and the local field potentials. So these things, the relation between these three is not so straightforward as you may think. So basically at the causal chain, it all starts with some spike, some neuron that comes through the axon terminal, releases some neurotransmitter, and then causes asynaptic current, right? It's just a change in the conductance, which both can be excitatory, inhibitory, driving the LFP. So, you know, the LFP reflects both excitatory and inhibitory currents. And these synaptic currents, when they come in through the, the passive cable equation, they will cause transmembrane currents to flow through the neuron. So basically current exiting, completing sort of the circuit. And those transmembrane currents cause return currents that complete the circuit in the extracellular space that then, due to the morphology of pyramidal neurons, set up dipoles. So you have opposite extracellular voltages here, and those, return currents, they go all the way up, through the skull. This is the ceiling, but imagine that's the skull. And that's what you measure, right? So when we stick in an electrode, then this is the LFP locally, and then macroscopically it would be the EEG, MEG. So the LFP, it correlates to synaptic potentials, but a little bit indirectly, right? The LFP itself is not really causative to anything. Okay? So it might have some effect on the neurons, but it's not strong. So it is an indirect proxy of causing the spikes, but it's also a consequence of the spikes, right? So that makes it hard to interpret, right? It's both a cause and a consequence, right? And then one thing is that the LFP, due to its nature, because it reflects these dipoles, it needs necessarily correlated synaptic inputs to arrive, right? So at any frequency, to even measure power, to measure a signal, the incoming signals need to be correlated with each other, right? Otherwise, you don't have a strong signal. So it's always biased to measure already correlated activity to begin with, which we will see has some implications. And then also it measures mostly the power at the lower frequencies. Right? It's biased to low frequencies, and the reason for that is that the neuron is a filter. The dendrites make it basically a low-pass filter, because the neuron responds mostly to low frequencies rather than high frequencies. So these causal relationships are generally complex, and you need to understand them to interpret functional connectivity measures. And now we can measure these signals at many scales. So we're going to today focus on LFPs, or when they're measured from the dura in patients, IEEGs, or EEGs when they're measured from the scalp. And so the size of a node, when we talk about functional connectivity, is always going to depend on your measure, right? So when you measure an LFP, then the node is sort of a few millimeters, and when you go to the EEG, you sort of have a maximum resolution, so it's going to be perhaps on the order of a centimeter. But the resolution is going to change how you process the data. So with EEG MEG, we have various techniques like beamforming or computing second derivatives, Laplacians, that improve your resolution. And then also the size of the node will depend on the correlations. So it's been shown, for example, by Gotha Einevoll, that if you have correlated activity in a local region, then the local field, the electric field, will already integrate over a larger region in space. Okay, so the nodes can be up to 5 millimeter large, and that's sort of a minimum resolution you can achieve with any of these techniques, I would say. So in general, and that's important, when we talk about a node in a network, it's not really clear whether we should think about these as areas, right? So they are measurements of electric fields, but the relationship between neural activity is local neural activity is not so straightforward. Even though we call it a local field potential, the issue is that the LFP is generated by inputs. And these inputs come both from local recurrent connections in the cerebral cortex, but also from efferent connections from other regions, right? So if we say, oh, this is the V1 LFP, well, that V1 LFP also contains inputs from LGN, thalamus, V2, etc. So the local field potential is not as local as its name suggests. So as nicely introduced by the previous speaker, we have sort of 3 levels of connectivity. The first one is structural. Structural is just the anatomical strength of connections that you would measure with a retrograde tracer, for example, so to get area level connectivity measures. And then functional is anything statistical, right? It's just a observation of statistical dependencies, whether linear or nonlinear, between 2 measurements of 2 signals. And then a third definition, which I think is a lot more fuzzy, is effective connectivity. And effective connectivity, I think Friston has defined it sort of as the simplest circuit or the underlying parameters of the circuit model that give rise, sort of in the generative sense, to your functional connectivity data. So the notion is here that your anatomical connections, for example, the synaptic density, will of course be the strength of the connections is both a function of the structure, but also of things like neuromodulators, synaptic dynamics, and what have you, right? And so that's sort of thought as the effective connectivity. And so you could think of techniques like dynamic causal modeling to infer that. And I'll talk a bit later in the lecture, how useful that concept actually is. Because we believe that it's actually mostly reflecting the linear aspect of transmission between brain regions. So in these 2 papers that give an overview of many of these measures, we talk about 2 fundamental problems with EEG-MEG. And one of them is the classic volume conduction problem. The volume conduction problem is simply the fact that through the tissue you have a local conduction of these dipole currents, the return currents, and so that means there is a necessary smearing that's of course much larger when you measure scalp EEG-MEG. Right? And this means that the LFP can be considered as some linear superposition or mixture from volume conducted sources that are both local and non-local. Luckily, these occur at instantaneous delay with EEG-MEG because the tissue is essentially ohmic from a physics perspective. And so there are various measures, some of them that we developed, that can address this volume conduction issue. And in recent years, we and, for example, Bujaki have been talking about another kind of problem which we call the synaptic mixing problem, which is another problem that I just referred to, and that's the fact that the LFP is a linear mixture of synaptic inputs from both local and non-local sources. And the issue here is that this occurs actually at delays, all right? So now the question is, how can we unpack these signals, functional relationships, to talk about effective connectivity again? So I'll explain this problem in more detail. So here's a general overview of functional connectivity measures used in EEG-MEG. I generally sort of look at them in 4 axes. So the first one, are they frequency resolved, right? Some measures, they live in frequency space, so they're directly spectrally resolved like coherence. But there are examples of just simply Pearson correlation that is not. And then a very important distinction is between linear measures and nonlinear measures, right? So linear measure will capture basically just transmission, and so it's just a scaling or a phase shifting. Nonlinear measure express anything that has higher order relationships. And so various measures like transfer entropy capture also nonlinear dependencies. And then another important distinction, is your measure directional or not? And then fourth, does it deal for EEG, MEG with this volume conduction problem, right? So a lot of studies use, for example, coherence. You know, it doesn't really deal with volume conduction, so people use things like weighted phase-lag index or imaginary coherence, but these are still approximately linear measures. Things like Granger, you know, are directional, but still they are influenced by volume conductions. And then these kind of measures, we'll talk a little bit about them, like synergy, redundancy, transfer entropy, weighted symbolic mutual information, are nonlinear measures that are usually not frequency resolved, and some of them deal with volume conduction. So when you use a measure, think of these four axes. All right. So let me see. Okay. Yeah, so coherence, let's start with that. That's one of the most widely used measures, as nicely introduced for EEG, MEG. And there are several measures that are very related to that, like phase-locking value, but they have very similar properties. So coherence is just a linear measure of explained variance in the frequency domain. It literally tells you how much variance you can explain at a given frequency in one signal by another. So it only has the relationship at the same frequency. And there are two ways in which coherence can change between two signals. You can either have a change in the phase relationship between two signals, right? So it's a phase shift, but the coherence might stay the same. Or here you have two cases where two signals change the relative coherence strength from 0.1 to 0.8. And probably the most common interpretation in the field of EEG, MEG is to take these measures of coherence and look at them directly as some form of synchronization between oscillators, or they're interpreted as a measure of effective communication. Right? That has been proposed, for example, by Francisco Varela, Steve Bressler, and Pascal Fries. And so the idea here is that you need some form of mechanisms to communicate flexibly between brain regions. And it was proposed that if you have oscillators, you can change this phase relationship between the oscillators, so the synchronization between different oscillators, and thereby, for example, in this case, between the red and the green area, you would have the signals coming in at periods where the neurons are excitable, okay? Whereas with the black population, the signals would arrive when the population is not excitable. And there is some evidence that there are coherence changes with cognitive functions, for example, like attention, right? So as you see here, with attention inside the receptive field, the coherence between V1 and V4, it increases as compared to no attention, right? And that was seen as sort of evidence for this theory that maybe attention, effective communication is established by having coherence relationship. So this is kind of interesting because here, the functional connectivity is actually taken directly as a mechanism, as sort of a causal entity in the causal chain, right? And we have criticized that idea because it sort of supposes here that coherence is the cause of the communication itself. But it's of course obvious that any coherence must somehow result from communication itself, right? And so we wondered actually, can we explain the coherence from the communication between the neural nodes in the first place? And in that sense, I will show you a model that suggests that yes, we can. And in that case, the coherence doesn't really indicate a functional consequence, but we can infer the effective connectivity from it. So the idea is very simple. If you have a sending area, that sending area will project to another area with some effective connectivity parameter W. And due to the nature of the LFP, the signal here is going to be some superposition of the incoming signal with the intrinsic fluctuations in some receiving area. And that's the measured signal Z2 here. And so you would have your sending signal Z1. And because you're measuring essentially the same signal in two places, right? Because it's just a delayed copy, you're going to have interareal coherence that occurs at a non-zero phase delay, right? And if you now mathematically analyze that, which is what we do, you can derive a very nice mathematical expression for the coherence where you can show it's proportional to the effective connection weight. And then it's the power in one sending area, so at a given frequency. So the power spectrum here relative to the power spectrum in the receiver, and that ratio strongly drives the coherence. That means that if you have a sending area that has a lot of power at beta frequency here, and the receiver doesn't, then you will see by definition that the coherent spectrum shows a peak at that frequency, right? It doesn't mean that you have synchronization at that frequency, all right? It's just a consequence of, it just tells you actually something about the power spectrum of the source. So that's very important because there's a lot of confusion about it in the EEG-MEG field, because normally people look at coherence measure like that and say, oh, this means there is an oscillator in this area 1 and an oscillator in area 2 that is synchronized. So that's not the case because the intrinsic activity here is completely uncorrelated. I refer to the paper because there are some more intricacies to this model, because you also can refine it by knowing the number of projection neurons in a given area. So we have solved this mathematical problem, and we have applied this to various systems to predict the coherence in a given moment and then infer the actual effective connectivity parameters. So here's an example of monkey recordings of local field potentials between two areas of the premotor and the parietal cortex while the monkey performs a visually guided memory task, right? And you see here that the coherence changes a lot between the different task periods. But we can fully model that actually based on the changes in the power in a sending region, on the beta power, and now we can infer the effective connectivity weights in this model that actually do not change between the different task conditions, despite the fact that the coherence seems to indicate a massive change in the functional connectivity. All right. We've also shown that the anatomical structural connectivity shows generally, as predicted, a very strong correlation with the functional measurements of LFPs. This is data from ECOG and monkeys. So we can explain about 42% of functional connectivity based on the structural connectivity in a linear manner. Right. And another thing that we can model is this idea that communication in different directions occurs through different bands. So it's thought that feedback communication happens more at alpha or beta frequencies, whereas feed-forward communication is thought to occur more at gamma frequencies, and we've shown that this can be actually explained by the presence of the power in lower and intermediate areas of the cortex. So it doesn't actually reflect a preferential communication at those frequencies. It merely indicates the distribution of power. Then another important point is that in general, LFP, due to its nature, LFP coherence doesn't really have a good correspondence to what happens at the level of spiking, all right? So this is an example where you see coherence between the LFPs from two regions, but when we now look at the spikes at the same frequency, we actually see that the spikes in the receiving area in premotor cortex, they are not phase-locked to the fluctuations in the parietal cortex. So it means that the coherence reflects sub-threshold relationships, but not necessarily super-threshold relationships. And that's also quite different from how people have interpreted, and that's something we have tested also with optogenetics. So we looked at cases where we have coherence, and then we use optogenetics to silence the spiking activity of the excitatory cells in one area while recording with neuropixels LFPs from both areas. And in that case, we could show that the coherence exists despite the fact that we completely silence the spiking in one area. So that, again, shows this non-local nature of the LFP. And it's also something that we have confirmed with computational models, that if you don't have resonance, spiking actually makes a small contribution to the LFP coherence. Is mostly driven by the synaptic inputs from one area to the other. So how many minutes do I have? 10? Yeah, okay. So just to wrap this part up, I would say that linear measures like coherence, they do not necessarily indicate synchronization at the process level, all right? So what they primarily indicate is a linear signal transfer from one area to the other that has something to do with the nature of the LFP. And in fMRI, because fMRI also reflects synaptic inputs, this kind of formalism also applies for fMRI, right? It hasn't really been applied, but it does actually apply to fMRI in a similar manner. So this means that coherence can be actually explained as a consequence of the communication, which actually is not so far off from how the people first using that term were interpreting it. But it itself as a statistical quantity doesn't have a causal consequence, all right? And the third is that I've showed you a model where you can infer effective connectivity from a linear functional connectivity. And the crucial thing there is that you have to take into account the power ratios between the areas. But we've also shown in bidirectional systems, I refer to these papers, that you also have to take into account phase delays, at least for higher frequencies in this model. And we've shown that this model has actually quite some advantages. It's much more interpretable than dynamic causal modeling. Now, as I said, so if you want to understand the brain, so if you come more from a systems neuroscience perspective, linear measures might be useful to infer effective connectivity, but they're not necessarily useful to actually study how neurons compute, all right? So if you generally think about a computation, then you have some sender region that sends some spikes with some effective coupling weight, right? So this would actually be your effective connectivity. But, you know, effective connectivity doesn't really tell you how a receiver integrates or transforms those inputs into the outputs, which is something you actually want to measure and know, right? And there's two aspects of that. So first, there could be some kind of resonance. It means that a receiver preferentially responds to some frequencies and not to others. And then there will be always some computation in the local recurrent circuit that amplifies certain patterns in a highly nonlinear sense, right? And then, of course, these two regions are recurrently coupled, so it becomes even more complex. Now, nonlinear transformations are crucial to understand any computation in the cerebral cortex. And the reason for that is that all of the patterns that our brain recognizes initially are very nonlinearly entangled. So if you, for example, think of the picture of a dog, you could see that picture in any different kind of pose, shape, size, right? But you are able to recognize it, and we know actually that at the higher levels of the brain, there are linear separation planes between these two classes. But initially, these dog pictures, when they fall in your retina, have completely different poses, etc. So they're extremely nonlinearly separable. So that means that the transformation between areas have to be very nonlinear, as we know from deep neural networks, to create this linear separation, to make this information accessible at higher levels, all right? So linear measures cannot do that because they can only relay information. A linear measure cannot actually process something, can only relay. And there are generally three forms of sort of operations in neural networks: pattern completion, where you start from two different images of, let's say, a dog, and you map them onto the same concept. But you also have pattern separation, which is a very different operation, where, for example, you have two wines that are very similar, but you want to distinguish them. And then third, selection. So how do you actually select one out of many input patterns, depending on cognitive conditions? So we have shown, for example, that attention, there's a recent modeling study, can be resolved purely through nonlinear dynamics in a recurrent network. So if a recurrent neural network gets inputs from multiple streams that are competing, and the network has to actually select one of them based on top-down feedback, we have shown that in this case, top-down can, through the nonlinear dynamics in a recurrent network, completely bias it to process only one of the two inputs. And then this is quite interesting because in this case, the effective connectivity is identical, all right? But the area actually responds only to one of the two inputs. So that's something that linear measures would not pick up. And when you look at population dynamics of the spikes, then you see that the trajectories are very different. So coherence is a measure that only looks at one frequency, the same frequency at a time. Nonlinear measures look at different frequencies, all right? So the nonlinearities always transform energy from one frequency to other frequencies. And here, just to give you an example of a nonlinear measure, this is weighted symbolic mutual information. So in this case, in the linear measure, you look at basically linear mappings of small pieces of the signal in one area to another area. And here you actually look at relationships that are not a linear scaling, all right? And these measures in general can have superior sensitivity to states, cognitive states, and for example, also pathological states compared to linear methods. So in general, the difference between linear and nonlinear measures is summarized here. So if you want to infer effective connectivity, then linear measures are very useful, as we've shown, but nonlinear measures are not useful, probably because indirect links can produce strong nonlinear relationships. All right. Volume conduction strongly affects linear measures because volume conduction is linear, but it weakly affects nonlinear measures. Linear measures will mostly affect subthreshold activity. Nonlinear measures will affect mostly superthreshold activity because the conversion of a spike, inputs to spiking is nonlinear. And if you want to infer transformation of inputs to outputs, then nonlinear measures will reflect more the integration. And here I give an example of a study, for example, where a linear measure or closely, approximately linear measure like weighted phase-lag index does not indicate whether participants, for example, bind two auditory streams into their consciousness, whereas nonlinear measures here, they do strongly indicate if subjects are binding two auditory streams into one sound, right? And there are various examples of this also in ground truth simulations. Then there is another aspect that can be quite interesting also for fMRI or PET, which is looking at redundancy and synergy based on information theory. So if you observe covariance between signals, then that covariance can actually express both redundant or synergistic relationships, right? Redundancy is generally something that is characteristic for linear transmission between signals. But if you have a recurrent nonlinear system, then you can have the emergence of synergistic relationships between areas where if you observe two signals together, you extract more information than the sum that you can extract from either one of them, all right? And this actually means that the system can now only be sort of examined holistically because you cannot partition it anymore into nodes that separately convey information, because the information actually lies in the entire pattern. And we have shown recently in this paper in press that, for example, the ERP signals, the event-related potentials, or the broadband fluctuations from the cortex of marmoset monkeys convey actually highly synergistic information about, for example, sensory prediction errors. So observing both the frontal cortex and the temporal cortex together gives you synergistic information rather than redundant information. So to wrap this up, to conclude a little bit, so I've shown a little bit our model, the synaptic source mixing model, where you've seen that you can infer linear functional connectivity. Sorry, you can infer effective connectivity from linear functional connectivity, but that effective connectivity, sorry, the functional connectivity like coherence doesn't necessarily indicate a functional consequence. The coherence should not be directly interpreted in terms of synchrony or effective communication. And second, that nonlinear functional connectivity measures can be highly diagnostic sensitive at different states, and they have various properties that are complementary to these linear measures. So for more reading, I refer to these two overview papers on the topic. And with that, I would like to thank the people in the lab that have done this work, and some of our collaborators on monkey data, like Alex Thiele, Hans Scherberger, Wolf Zinger, and then also Andres Canales Johnson from Cambridge University. Thank you very much.
Great, thank you. Thank you, Martin. A beautiful summary of really adjacent fields that, yeah, from imaging we usually don't know that and think about that. And I very much appreciate that you mentioned the problem with areas being not automatically the senders also, and you don't know where the input is coming from. So this is very important that we have to keep in mind for macro scale imaging, I guess. Is there one pressing question already in the audience that knows that they want to ask something? Otherwise, I'll ask Simon up there. You're kind of floating above us there, like the god of connectivity.
Yeah.
So have you found any online question that is interesting to pose here?
Not really, but
because we are,
maybe
yeah,
I will ask.
okay.
Yeah, but maybe just ask to give a thought, an initial impression on something that we will talk about a lot later: statistical versus neurobiological definitions, because it became quite clear in the first two talks that we as the entire field seem to undulate a bit between defining connectivity more neurobiologically, physiologically, or more statistically. And just give us your initial 1 minute on what's the pros and cons in both, and where do we want to go?
I think that was for you, Martin. now.
I have to.
Can we prepare already the next slides, maybe,
Yeah.
for Lucina?
So I think in general there is way too little work to actually validate that relationship, right? So I think, for example, I don't think the solution like dynamic causal modeling that gives you the full answer to that. So I think we need much more work to validate, and so for that you need actually causal perturbations, and you need ground truth modeling studies of spiking recurrent neural networks where you can actually do that, so that you can strengthen the link. And you can actually see what kind of functional connectivity measures are actually useful to characterize certain cognitive processes, right? So that's sort of our approach. So I think there is a lot to explore actually at the functional level, using many measures that are also nonlinear, that are really underexplored, and that can be also clinically very relevant without necessarily having to interpret the underlying neurobiology. And at the same time, I think the linear measures like coherence have been massively overinterpreted, and so there is a lot of work to be done there. That's my 1 minute.
Thanks.
Great, thank you. Thank you, Martin.
That's one question, I don't know.
Yeah, great. Can you, wait, I give you the mic. Otherwise we don't hear you online, I guess.
So since the linear measures and nonlinear measures are basically operating at different domains, like suprathreshold and subthreshold, is it possible that the Granger causality, for example, points from one region to another, so that the direction is pointing one way in the causal measure, and if you use a nonlinear measure, the direction is reversed? So
that's, yeah, exactly. That's absolutely possible, right? Because you, so the linear measure, we have some examples of that where the linear measure is actually strongly biased because one area has strong power. And then if you actually look at the actual integration, so the nonlinear interactions, they can actually occur in a different direction. So it's exactly that, because the Granger is, as coherence, very biased by just the presence of power in one source, right?
Thank you. Yeah, perfect timing now that we have. Please give another round of applause to Martin for his contribution. And then it's a great pleasure for me now to introduce the next speaker, who I guess has traveled quite far to reach us here. It's great to have you here, Lucina. Lucina Udin is a professor and directs the Brain Connectivity and Cognition Laboratory at the Center for Cognitive Neuroscience Analysis Core in the Semel Institute for Neuroscience and Human Behavior at UCLA. And within a cognitive neuroscience framework, Lucina's research combines fMRI, functional and instructional neuroimaging, to examine the organization of large-scale brain networks, which support the development of social cognition and executive functions. And yeah, I'm very interested in what you're going to present us today. I hope the presentation is working. Simon, are you seeing everything online?
Perfect.
Great.
Well, Lucina, good to see you, even just virtually.
Okay, great. Thank you so much. Thanks to the organizers for inviting me. It's my first PET conference, so great milestone for myself. And also, thanks, Valentin, for moderating this session. I don't know if you remember, I was here 10 years ago for the first time giving a talk, and you took me for pretzels and beer over in the Wiener Platz. So it's nice to be back after so long. And also, I wanted to say I really enjoyed this presentation from Dr. Ariana Sala this morning, because I learned so much of the history of connectivity, and I'm embarrassed now to find that I was unaware of some of this early work. Really embarrassing to, as I spent 20 years studying brain connectivity, I was always looking at it through an fMRI lens, and so for me, it starts in 1995, but really these concepts have been around for a long time. It shows us that maybe we can all do some reading in our spare time. So thanks so much for this. So I'll talk today about this problem of nomenclature. Especially it comes from fMRI because I think we've seen such an explosion of fMRI functional connectivity since 1995 or 2000. It's too much, really, like thousands of studies, right, that use resting state functional connectivity. And this explosion, I think, happened before we could even understand what is connectivity, what are we measuring, what should we call it, what does it mean. I think the field, the empirical work preceded the theoretical work in fMRI a little bit. So we're stuck now with these problems. I'll try to talk a little bit about what we've been thinking through as we wade through this explosion together. Well, of course, the slides won't advance. Oh, okay, great. So this is just a lovely picture of a person in the scanner doing nothing at all, what we call resting state fMRI. And of course, now we see the activity is not random, but the spontaneous activity that we can detect in the brain, shows this low frequency oscillatory pattern that recapitulates a lot of the networks we know to be involved in cognition. Of course, visual, auditory, sensory motor, attention, language networks, all of these things can be mapped using resting state fMRI. And there seems to be this spontaneous coherent fluctuation, which maybe everyone already knew from EEG, or they already knew from these other measures we heard about this morning. But I think it became revolutionary for fMRI people to realize, oh yes, actually there's some structure to spontaneous activity in the brain, and especially nowadays we call it connectivity, but really it's statistical correlation between the signals in discrete brain areas. So I'm sure we'll come back to this definition of connectivity later on. What I will do is sidestep some of the issues. What is connectivity? The philosophy. I think I'll go straight to some of the problems. I'll also sidestep the interesting idea of effective connectivity and causation that our previous speaker just really elegantly described. When it comes to fMRI, there's these issues. We all know correlation is not causation, of course, but we always try to kind of infer some kind of relationship between areas, even from fMRI, as coarse of a method as it is. So there's a review here where we try to talk through some of these issues, but it's an issue I'll sidestep again in favor of talking today more about the nomenclature problem. Okay, so we're gonna do a game. It's audience participation. Please yell out, just tell me, what is the name of this network? Okay, so everyone, or many people are confident that this is a visual network. Okay, let's try another one. Please name this network. Oh, even more consensus. It's very, very clear. Everybody agrees this is default mode network. Okay, let's try again. Please name this network. Language. Any other guesses? Executive. Task positive. So, okay, so there's some more mumbling. Okay, we'll take that as lack of consensus. And one more. Silence. Some guesses? Wow, really? No? Okay. So we get harder and harder as we go along. So I think I'm just trying to illustrate the fact that we think we know what we mean when we say, oh, this is this network, this is that network. But I think there's a lot of intuition and guesswork that goes into this naming, and perhaps indicative of the fact that maybe we haven't quite defined what these things are. But we think that we're talking the same language. We think we have some nomenclature, but do we really? I mean, so what's in a name, right? There's papers that talk about and define and discuss what networks are. Here's one from Dosenbach 2008 saying, okay, a singular opercular network includes such and such nodes across the brain. I've written papers where I say, okay, there's a network called the salience network with such and such nodes. Others have, for a long time in the functional MRI literature, talked about core regions of a certain network, ventral attention. All of these, if you look at them, they overlap in a lot of the brain regions that they include. So all of these regions, all of these network nomenclature schemes include the anterior insula cortex, even though we're using very different terms now. Where Dosenbach is saying, well, this is a singular opercular network, and I'm saying, well, it's a salience network. And Corbetta as saying, well, this is obviously the ventral attention network. So I think we've been using these words, we've been using the terminology, even though we haven't exactly said what is it we mean by this network, what brain regions does it include, what's the scope of that description. For the insula in particular is my favorite brain region, but I think this is problematic because if you use a name and it doesn't refer to the same thing every time, we're really just introducing a lot of noise into the field. Here's 4 very popular parcellation schemes that different scientists have created: Gordon, Yo, Shen, and Shire. And many people use these atlases to refer to their own findings and say, okay, my findings map onto this atlas and this name. So if you are using these 4 different atlases, you might just find really different descriptions, and you do in fact, of the same network, which is called the salience network in each of these, or salience/ventral attention if you're Thomas Yo and you like to make it more interesting. But in any case, if you overlap your own findings onto any of these regions, you might get a different answer to the question, do my findings correspond to the salience network? And I think this is bad for the field, really. If we can't decide what the networks are called, how can we evaluate new results, look across labs to find convergence? I think the worst implication of all of this is in the clinical domain, because people are saying, well, we're going to target this region with TMS. We're going to do deep brain stimulation of this node. I mean, these are really important clinical questions. And if we can't agree what to call something, it's really hopeless, I think, when we get into the translation. So I think if not for our purposes, for the clinicians, we have to do something a little bit more concrete in terms of our nomenclature. And so we've been thinking about this problem of consensus, convergence, correspondence among functional brain network atlases. All of this, of course, from resting state fMRI, only because it's such a widely used approach these days that we sort of can't avoid it. A lot of the work I've done in my lab and with collaborators in the past has been thinking about network dynamics and what we've called these areas and how they interact amongst each other. I think I'll just skip this for the interest of time. But I think when you look at the insular cortex in particular, it seems to be involved in very, this is from a different slide, but anyway, ignore that pain. Yeah, but anyway, this area seems to be involved in emotion, pain, awareness, interoception, somatosensory processing, but also high-level things like conflict, attention, switching, inhibition. So almost every kind of imaginable cognitive term you can use has been implicated with insular activation. And this might not be so surprising because this area shows a lot of functional flexibility. So across different tasks and experiments, the anterior insula shows a high level of activation, if you use meta-analytic kinds of approaches to look and see. There's a lot of flexibility associated with this brain region in that it participates in a lot of different cognitive components. And also, there's a lot of functional diversity to this region. There's subdivisions, of course, anterior, ventral, anterior, posterior. And if you look at, again, using meta-analytic connectivity mapping or other such approaches, there's a high degree of variance in what the insular subdivisions are doing, especially the dorsal anterior insula, which shows a great deal of diversity in terms of the cognitive functions that it's associated with. So maybe it's not surprising that different network schemes have added this brain region to different networks because it's just involved in so many different things. It's also very interesting in terms of its dynamic functional connectivity or changes in functional connectivity over time. So instead of looking at like the average correlation pattern between the insula and the rest of the brain, if you break it up into, let's say 45-second windows or 60-second windows and use these dynamic approaches where you can look at frequency of occurrence of a specific brain state or dwell time of a brain state or transitions between states, and look at the insular cortex in particular, there are some states in which all these different subdivisions are talking to all the other brain areas at about roughly the same kind of frequency, and other brain states that occur less frequently where the subdivisions are very different in their connectivity patterns. And I know this is impossible to read, but I just wanted to show the point here that the dorsal anterior insular subdivision, which is the one in red at the top, is the most diverse in terms of its functional connectivity pattern. So between different connectivity states, it shows high or low patterns of functional connectivity with the rest of the brain, and the states are all different from each other, whereas the other subdivisions seem to be more similar in their patterns of whole brain functional connectivity. So this is all just to say we know that these problems of network definition are confusing and complicated, and there won't be straightforward answers because each of the brain regions we might think about placing in one network has this like multiplicity of function and very diverse activation profile and connectivity profile and pattern of dynamics. So maybe it's not surprising that we haven't yet reached a consensus on some of these things. So the questions that I am trying to explore now with the group are how is a functional brain network defined using fMRI or resting state fMRI in this case, and how many functional brain networks are there? And I don't think we'll have any consensus here at all. So, but we still have to pose the questions, I think, as a starting point. So as this starting point, I got together Nathan Sprang and Thomas Yeo, and in 2019, we started to think about the issue of could we arrive at a universal taxonomy of macro scale functional human brain networks? And we thought, let's give it a try, right? We'll take a first pass at this question. And so if you think about some of these, basically the atlases, networks, or independent component analysis parcellation schemes, there you do see some things that come about in a lot of different schemes. Like we talked about the visual network, many of you were very happy to say, okay, that's a visual network, something in the back of the brain, occipital. In the Gordon parcellation, it's the blue patch there in Steve Smith's independent component analysis. It comes out again as a medial and lateral visual. In Thomas Yeo's parcellation scheme, again, you see these purple and red in the back of the brain. So there's some things that are not so controversial, and maybe we can say, okay, we have some idea of what that is. And when you look at things like the motor cortices, again, you can find these in independent component analysis types of studies. You see a motor or somatomotor patch in most parcellation schemes. Again, I didn't go into the detail here, but many of these parcellations are based on resting state fMRI and trying to find areas where the functional connectivity patterns change abruptly, and we call those the borders of where one network ends and the other begins. So again, these are very rough, coarse measures of connectivity. So you can see some consistence, like blue in this Yeo network, Yeo parcellation scheme. And in Gordon, you actually see some subdivision between a hand motor, a face, a foot, and various other. So, motor cortices maybe not so problematic, or sensory motor. But, and then we start to get into those networks where people are more unsure and they're more mumbly in their responses. So when you think about things like a dorsal frontoparietal network, Corbetta has called this a dorsal attention network, and so have many others. That comes up in many different meta-analyses and in resting state parcellation network atlases like the Yeo and Gordon. It's green, actually, nicely convergent across those two networks. It's green in both of those papers. So maybe it's not so bad, but we start to get into these things that some people like to call executive networks or cognitive control networks, frontoparietal control networks, multiple demand networks. These ones really get a lot of names. And you see that if you try to look across the literature a bit. It's orange at the top in the Yeo networks, and I'm showing a couple meta-analyses on the left, which give them different names. And the Gordon one breaks it down into a frontoparietal and a parietal memory. So we're getting a little more complicated and a little more lack of consensus as we move from the more, sort of basic sensory processing into the more complicated cognitive functions. This is the worst one. This is also my favorite. The salience network, cingulopercular network, ventral attention network, whatever you want to call it. It's shows up in a lot of studies. I think the salience network nomenclature came from a Seeley et al. Paper in 2007 using independent component analysis on resting state fMRI to break apart the task positive network into both a salience and an executive, what they called it. Corbetta, of course, predated all of that. This is just a review that talks about the ventral attention network that's actually right lateralized, so that's another story. But yeah, the Yeo and the Gordon networks have diverged in their naming. Of course, Yo calls it salience ventral attention, and Gordon has a separate singulo-opercular and a separate salience network with a lot of overlapping territory and a separate ventral attention network. So, you know, some people think they're three different dissociable networks, and that also we have not reached any convergence on as far as I can tell. And this, of course, everyone likes to call the default mode network, and nobody has any problem with that. So maybe I should leave it alone. But still, I think it's nice if we can get at what I think is some kind of terminology that we can all accept and is based on something like anatomy, which perhaps there can be more agreement on than functional significance. So, and the truth is, even if you believe there's a default mode network, there's subdivisions, and maybe a medial temporal lobe subdivision and other some areas like hippocampus. Does that belong in default or doesn't it? It depends on who you ask. I don't think it's quite as straightforward as maybe our initial poll indicated. So we tried to propose this taxonomy saying, well, let's anchor it in anatomy, let's anchor it in things we all agree on. Let's just start simple and say, we'll take the cortex and say, there's an occipital, it's in the back of the brain. People tend to call it visual, but if we stick to the anatomical terminology, it might be easier going forward. A pericentral, which involves somatomotor cortices, a dorsal frontoparietal, which some people call attention network, a lateral frontoparietal, which some people call a control or executive control network, a mid-cingulo-insular, which again, salience and many other names, and then a medial frontoparietal, which of course is happily referred to as default network by many. I cannot tell you what a failure this was because almost nobody adopted this nomenclature except me. I just started using it in all of my papers, and almost, well, maybe a couple of you did, and for you, I thank you. But I think it's very hard to change the trends, or it's hard to change the culture after many years of calling something one way. So a very astute reviewer said, I think you should form a committee to form a consensus around this issue and get buy-in from many people in the field. It did, yeah, like I said, it didn't work because many reasons this nomenclature didn't work. We didn't, think very clearly about dynamics, as I mentioned, inter-individual variability, subcortical brain regions, fractionation and subsystems and hierarchy. So we took the advice of the reviewer who said, why don't you form a committee? And Anastasia Yandiki, who's sitting here, is part of that committee, and she came up with the wonderful name of WhatNet, the work group for harmonized taxonomy of networks. And I think that's our greatest contribution to the field is this acronym so far. And so the committee is now endorsed by the Organization for Human Brain Mapping as a best practices committee. Are some of you familiar with the best practices committee on data analysis and sharing? That's called COBIDAS. They also recently published, a few years ago, published a paper on MEEG best practices. And so we started this committee on best practices, which is now called WhatNet. And we published a long paper talking about these controversies and progress. This is now in Network Neuroscience 2023. That was the preprint there. In that paper, we did the survey that I just showed you at the beginning, and there was a lot of consensus for default network and visual network and motor network. People just immediately said, okay, that's the name, but really no consensus, a very low convergence across the frontoparietal and cingulo-insular systems. As much, we might have predicted that. So here's the thing, we cannot change people's minds. We cannot undo the past and go back and recreate the atlases and give them consistent names. It's too late, right? The ship has sailed, and there's hundreds and hundreds and hundreds of papers that have used these nomenclatures from these atlases for better or for worse. So we don't have time to go back in time, or even if we could. Perhaps these atlases have served a function, which is just being reference points for us to take our findings and say, okay, here's how what we found in the lab maps onto Gordon's network. Here's how what we found in the lab maps onto Thomas's network, Yo network. The problem is, as I mentioned earlier, some of these networks are inconsistent themselves on their nomenclature schemes. They don't call the salience network the same cortical regions even. So it's not so great, right? We're still left with, okay, how do these things what's the convergence or what's the implication for some other field? So this is our latest attempt, maybe our final attempt at trying to address, probably my final attempt, because I'm tired. So this is what we did. So Ruby Kong in Thomas Yo's lab has developed a network correspondence toolbox, which we're about to release. So this is one example here where we included 16 widely used functional atlases that include networks in them and their labels. And we created a tool to easily map between them and to easily map your own results against them. And I'll just show you what that looks like a little bit in our final minutes here. So this is Ruby Kong, who spent last couple of years trying to develop this toolbox, and it's finally ready for release. So you can basically take any data file. Let's say you made a contrast between two conditions in a task fMRI paradigm, and you have a data file now. Or let's say you conducted independent component analysis with your resting state fMRI data set, and you have one component now. You want to say something about that component or something about that contrast. So you take your own data, and we have now, as I mentioned, 16 different atlases in the toolbox. And what that does is really simple, just takes a computation of the dice overlap between all the atlases in our toolbox and your input data. And it can give you some more details about that, but basically it'll show you the overlap between your input data and all the networks within all of the atlases that you select. So in your results section, you could now say something like, we found these findings, they overlap very high with what Yo network calls control and what Laumann network calls medial parietal, or something else entirely. But you could do this for all of your atlases so that you have a rough idea. If there is some divergence between a couple of atlases, you can look across all of them, so you're not just limited to one or two or three even. So this was just to show, since so many people tend to use these Gordon and Yo atlases, we just showed an example of what happens if you try to correspond the network names between the two. So for example, what Yo calls salience ventral attention A actually does map nicely onto, what he calls ventral attention B does map nicely onto what Gordon calls salience, but ventral attention A maps onto cingulo-opercular, if that makes sense. So you can see there's, and the stars indicate, we use spin tests to look at significance of overlap. So this lets you see between the two atlases, if you called it this or that, which one would it correspond to? I don't think this is an answer to our nomenclature problem. This is more of a Band-Aid, but I think it's a useful one because at least we know what we're talking about now. So if you took your own data, let's say now you have your own data and you're ready to go with the toolbox. This is an example. We just took the Human Connectome Project 2-back versus 0-back working memory contrast. So that's, you can see up there in the yellow on A. And you stick that into the toolbox and you say, let me compare it against all these atlases. On the bottom, you'll see this really nice plot that shows you just 4 networks, just for illustration here. So Glasser, Yo, Gordon, and Shen. That input data overlaps to a high degree with what's called frontoparietal in the Glasser network. Also, actually, with frontoparietal in the Shen and frontoparietal in Gordon. So you can report this in your results section. I think it's a little clearer than just saying, I think it's this network, that network. There's a network radar plot and some statistics you can do for a bunch of tables that you can also report to show the exact dice overlap, the exact p-value of significance of the spin test, et cetera. So a little more quantification of, rather than just my intuition is it's this network. And so this works in easy cases, like I mentioned, the Human Connectome Project 2-back sort of easily maps onto frontoparietal or dorsal attention across multiple networks. The strength is, the overlap is very high, so you can say with some kind of confidence, like my findings overlap with these networks across multiple atlases. This is a hard case. Let's just take a biobank, UK biobank ICA map, could be anything. But here, at least in this difficult case, you can say, well, it overlaps pretty high with cingulo-opercular in Gordon's atlas, or it overlaps pretty high with this, but clearly it's not consistent. You see lots of different networks are popping up here. Okay, I think that's it. Yeah. So I just wanted to acknowledge the OHBM best practices WhatNet committee and all of the folks who've been working with this, I guess now for 5 or 6 years. I gave you nothing really, but other than to illustrate that we have a problem. There's some possible paths forward, I think, in terms of getting towards consensus nomenclature. But this is, as I mentioned, just a solution in the fMRI domain. But I think you can input all different kinds of maps if you have them from different modalities. But of course, all these atlases were developed in the fMRI modality, so maybe they're more suited for that. But it's just a way of tackling, or a way of thinking about the problem as we've confronted it from OHBM and from the resting state fMRI end of things. I'm not confident that this will work either, but we had to try. So anyway, I'm happy to take questions now, and thank you all for your attention.
Thank you, Lucina. Oh, hands raising already. Beautiful summary, and particularly mentioning, I think, the critical problem that having a cognitive annotation to a network really implies a lot of functions that then maybe a clinician would use or so, and yeah, that's, I think, the most tricky part. So, Simon, sorry,
Yep,
you're
there's
second.
an
because
online
Aaron was
question
here
already
first,
in the chat. I will
this time.
direct.
So I'll briefly hand over the mic, then we can all hear you.
Thanks, Lucina. One question I have, particularly given the audience that we have here today, like this quite multimodal, how dependent do you think that these networks are on the imaging modality? So do you think that we'll see these in, say, PET imaging, EEG, and so forth? I have my own thoughts about this, but I'd love to hear yours.
Yeah, no, I do think they're dependent. That was sort of my caveat, that this might work reasonably well for fMRI new data. It might work to some extent for other modalities. We haven't tested that. There's different kinds of connectivity definitions were derived from different assumptions and all that. So I have no reason to think that this will work equally well, but it's more of like referencing and talking about things and reporting and clinical translation. That's where I think this will be better than what we're currently doing, which is, to me, a little bit guesswork. So yeah, I think it remains to be seen how well it actually maps on.
There's also a question in the chat that I would like to raise here. Where are where is the subcortical networks? So that's obviously a bit of an issue with all the parcellations. They're great for the cortex, but I think this question here by Jian Zhang puts the finger right into the soft spot. Subcortical, cerebellum, going all the way down to the brainstem. Do we have any chance to incorporate these as well?
Yeah, I'm going to blame the Atlas creators. So you should ask Thomas Yo, you should ask Evan Gordon, where are the subcortical? But the truth is there are now more subcortical network atlases that have been released. And I think one of them might be included in our toolbox, but the functionality of the toolbox we created is that you can upload a new atlas into it and you can use that as a reference atlas. So the idea is for the toolbox to be evolving and include new atlases. In fact, you could include a PET atlas, you could include, so I think the idea is that you can make it your own, but just so it's to facilitate the reporting and the comparison. So I think we can include subcortical atlases as they become available more and more, and we should really.
Lucina, thanks for the great talk. Obviously, there are rest and state networks that are more consistent and less consistent. Do you know, or do you expect that less consistent networks will be detected in a more consistent way with a better technology? For example, in using a 7 Tesla MRI. So in other words, it's a methodological issue, or it is a rather biological issue, so that there is a higher inter-individual variability for these less consistent networks, or it is both?
I think the problem I'm addressing is actually neither, right? Like it doesn't matter what let's say we have some ground truth of these are 10 networks and they exist, which is not true, but let's say we have a ground truth. I think the problem I'm talking about is we're calling them different things. So even if they were the exact same thing, different labs are referring to them with different names, and the nomenclatures they've derived, some of them come from function, right? Some of them come from some other principle, right? So even if we had ground truth, the problem I'm trying to address is why aren't we using the same language to talk about the ground truth? And we don't have that. But even if we did, why are we still talking about them as if they were different, functional? It doesn't help us to move the field forward if we don't know that these findings are relevant to our new findings here. So, so I don't know if some networks are more consistent than others. That's an empirical question. Some have more individual variability. For example, like the frontoparietal ones show more differentiation across individuals. That's definitely part of the problem, but I'm not even getting to that problem yet.
Great, thank you. Simon, do you have one last pressing question?
Well, there is one online.
Yeah, go ahead.
How valid are these networks, which are basically fMRI defined, in simpler mammalian brains, such as primates? I would reframe this question a little bit. Are we ready to include macaque atlases that may be constructed using similar methods as we have for humans? I don't think many people have done that yet. But do you see any fundamental problems in using the same methodology as Gordon and Yeo and all the others used, also in, let's say, macaques?
Yeah, I think in principle, of course, all of this can be and has been done in macaque. And so there's a PNS paper about the rat brain has a default mode network. There's another PNS paper about macaque default mode network. So clearly, like, you can use resting state fMRI on these various mammals, and you can find something that looks spatially similar to what people have called these other networks. I don't know if there's a lot of atlases yet out there, but I think in principle, let's say there's a macaque atlas, someone has developed it and validated it. In principle, we can use this toolbox for future researchers to benchmark their results against. So, I'm actually sidestepping all of these hard issues, like what is connectivity? Have we defined networks correct? I'm not giving you anything, right? I'm just saying, if we're going to talk in this language, maybe we can use common terminology. So I literally make no claims about whether these things exist across species or not. That's for the science to decide. But these are more about, let's at least report our results in a way that someone else can use them.
Great, thank you, Simon, for moderating that online, and thank you again, Lucina, for your contribution and for your really hard work to try to make sense out of that, or better to bring the people together. I think that's, as you said, it's not about finding a solution, but making people sensitive to this issue, which I think is very critical. So for the sake of time, we only lost like almost 5 minutes, so that's great, given all the hurdles in the beginning. I don't want to cut your coffee break now, so we make 15 minutes of break now, so we reconvene here at 15 past 11. So in 15 minutes for the online community, sharp, and yeah, enjoy your break. Okay. Hello. Does it work? Yeah. Simon, my major question. Can you hear me?
Yes,
Great.
and just as a minor thought, while I was hoping to be there until my flight got cancelled, actually, I think it's quite useful to have a second moderator that takes care of the online world. So that's
Indeed, yeah. I mean, it's a pity that you're not here in person, obviously, but yeah, I also thought it's kind of a nice back and forth. Makes sense.
made lemonade.
Yeah. Great. So as expected, we are late. Again, 5 minutes, but still that's doable. So we have 3 more talks now. One will be online, the second one from Young Hee. But now we'll have 3 more talks with brief discussion rounds afterwards. So if you have pressing questions, just raise your hands towards the end and I'll see you. Otherwise, Simon will take over again. Yeah, that's the competition now. Yeah, we'll start next. So first talk, Anastasia. Hello. So I'll briefly introduce the audience, you to the audience. So Anastasia Yandiki is Associate Professor at Harvard Medical School and an Associate Investigator at MGH. Her research focuses on computational methods for mapping the white matter axon bundles through which the different regions of the human brain communicate with each other. Her goal is to do this non-invasively to allow clinicians and researchers to study these connections in living human subjects. And her title now is very brief, Diffusion MRI. Thanks for enlightening us now.
Well, thank you for having me.
Do you have a mic? I think you need to use the mic.
Thank you for having me. Much better. I hope I can be coherent. As I was saying earlier, I woke up a little bit after midnight, refreshed and ready to start the day. So still going for now. So this has been a lot of fun and I'm already learning so much this morning. So now we're going to shift gears a little bit and go into imaging the structural connections in the brain with diffusion MRI. So most of what we know to this day about what is connected to what in the brain, and most of what a surgeon would know when they're about to stick a sharp object into your brain and try to figure out what they should and shouldn't cut, comes from 2 classical anatomic methods. One is gross dissection, as shown here, where an anatomist physically peels off fiber bundles in a fixed brain. This has rather low precision. It's really hard to follow individual fibers all the way from their origin to the termination point. And it's pretty low throughput. So it's hard to do even one single brain. It's very time consuming. And it is hard to produce a full connectome from even one brain. So as some fiber bundles are reconstructed, others have to be destructed. But what it has going for it is that it is feasible to do in a human brain. And so a lot of what we know about the main highways of the brain that we expect will be there in every individual come originally from this kind of study. And then the other, is it done already? My time is up. The other classical anatomic technique is tracer injection studies in non-human primates. So here we have very high precision, so we can visualize every single or individual fibers all the way from an injection point to all the parts of the brain that that injection site connects to. However, this is also low throughput. So with those classical techniques, it's not possible to do more than a handful injections in a single brain. It's a very kind of costly and time-consuming study to get from that surgery to figuring out what is connected to what. And also, it's not feasible in human volunteers. However, these are our 2 main sources of information about the ground truth of what is connected to what. So obviously there was a lot of excitement when diffusion MRI was introduced because it is very high throughput. So in just a few minutes, you can get a whole brain connectivity diagram from an individual, and you can do it in thousands or even tens of thousands of individuals, like some large initiatives are doing these days. And of course, it's also different in terms of compared to the other those anatomic techniques, because it's feasible not only directly in the human brain, but also in vivo. And so it's possible to follow individuals across the lifespan and look at different, the progression of disease, et cetera, and how it affects these connections. However, the big question mark is, what is the precision? So this is something that we've been trying as a field to quantify over the last couple of decades. So one issue that we are very aware of is that unlike those other techniques, this is not a direct visualization of fiber bundles. Instead, it's an indirect measurement of those fiber bundles. So the idea with diffusion MRI is that we're trying to differentiate tissues based on how water molecules diffuse through each type of tissue. And this diffusion is just random motion of water molecules. It has nothing to do with brain activity, and in fact, that's why we can do it in ex vivo brains as well. So the key difference between gray and white matter in terms of the diffusion of water molecules is that at the scale that we're imaging with MRI, of the scale of a couple of millimeters, the diffusion of water molecules is uniformly restricted in the gray matter, so it's equally easy or hard to move in any particular direction when you're in the gray matter. So this is isotropic diffusion. And in the white matter, the diffusion is anisotropic because it is preferentially restricted. So it's easier for, well, I don't know, should I use the online pointer? Probably. So it's easier for water molecules to move parallel to the fibers than perpendicular because of the presence of the myelin sheaths and the membranes of the axons. And so with diffusion MRI, we collect a bunch of measurements that tell us whether diffusion in a specific location is isotropic or anisotropic, so we know if we're in the white matter or not. And in different locations in the white matter, if we can infer that preferential direction of diffusion, the one in which it's easiest for water molecules to move, then we can infer the orientation of the underlying bundles. So it's an indirect measurement of bundle anatomy based on the diffusion of water molecules within and around the axons. So the way we collect those measurements is the scanner collects a series of images, and each one of those images tells us something about how much diffusion of water molecules there is in a particular direction. So you see these images here. The parts of the brain that show up as darker are the ones where there's a lot of diffusion in that specific direction, and the parts that are lighter are the ones where there's less diffusion in that direction. And then we collect another image that tells us the same thing for another direction, and so on and so forth, trying to sample all 3D space in all possible directions within the amount of time that we have. So from those images, we infer a couple of different things. Importantly, for imaging connectivity, we can infer that preferential direction of diffusion. So these vectors that you see here show us the direction in which there's most diffusion in every location in the brain. And if we follow those vectors around the brain and connect the dots, so to speak, this is what a tractography algorithm does. We can reconstruct the 3D shape of white matter tracts. And another thing that we can do from those same images is collect, infer a bunch of measures of tissue microstructure in each voxel, and there are many, many possibilities there, and it's a very active field, adding newer models every day. So some of the most known ones are the ones from diffusion tensor imaging, like diffusivity or fractional anisotropy measures. kurtosis measures or orientation dispersion, unit density from NADI, etc. So these are not measures that tell us something about what is connected to what. These are not informative on the existence of a connection, but some of these measures can be interpreted as a weight or a strength of a connection. So something like fractional anisotropy, to the extent that is a measure of myelination, for example, could be used as a measure of a weight or strength of a connection, because some of the underlying ground truth microstructural features like the axonal diameter or the myelin content could speak to, let's say, conductance, velocity, or something else that would be a real feature of the strength of a connection. So sometimes these measures, although they don't speak to the presence or absence of a connection, they are used as a measure of the strength of a specific connection that we reconstruct with tractography. So one thing of interest to this crowd is how do we get from those tracts to networks? And so the classical way to do this is to reduce the whole brain tractogram to a matrix based on the end-to-end connections between pairs of areas in a brain parcellation. So obviously it's highly dependent on which parcellation we would choose. And then once we have that matrix, it's treated just like a functional connectivity matrix, so it would be typically thresholded to reduce noise-induced connections, and then the same sort of graph analyses that you would perform on a matrix that came from functional correlations can be performed here to get different whole global features of the network, as well as to identify nodes with certain properties like hub nodes. And in terms of nomenclature, which is another topic of interest in this workshop, some of the nomenclature is pretty well established from anatomy. So in terms of the big highways of the brain, those have names that have been given to them by anatomists. So you see here on the left an image from the Schmamann and Pandya Fiber Pathways of the Brain book that shows a coronal section through a monkey brain. And you can see the main long association pathways, like the cingulum bundle, the superior longitudinal fasciculus 1, 2, and 3, middle longitudinal fasciculus, inferior longitudinal fasciculus. So those are pretty well-established terms to describe these pathways, although in some cases there can be some controversy or different ways of defining them, especially in terms of the exact endpoints of the connections. And so these main terms, these main names for the big highways of the brain have also been used a lot in tractography toolboxes. So as an example, what I'm showing here is the definition of these large highways that was produced by Chiara Maffei et al. And is part of our Tracula toolbox. So a lot of the focus of reconstructing connections from diffusion tractography has been these large highways. And we typically tend to think of them as an end-to-end connection, let's say between frontal and parietal for SLF, etc. But actually, the real picture is much more complicated than that. And if you go back to the anatomical texts and the source material for the definition of these pathways, you'll see there that there's an admission that actually they're not just a single end-to-end connection, but you should think of them as a highway with smaller bundles that are merging on and off and are actually carrying connections to multiple pairs of regions. So some of that detail and that fine print has been lost as we try to clean these images up and make them pretty for our readers and our users of our toolboxes. So some of the very early studies that compared these highways as reconstructed from diffusion tractography versus those source anatomical techniques showed that for the most part, diffusion tractography is doing a decent job reconstructing these large bundles. So this is an example at the top comparing the superior longitudinal fasciculus between macaque tracer studies and diffusion MRI in a macaque brain, or at the bottom, the inferior longitudinal fasciculus compared between diffusion tractography and gross dissection in a human brain. So this was the case even with the very crude methods, acquisition and analysis methods for diffusion MRI that we had, 15 years ago. But it also became clear very quickly that there was a lot of things that some of the fine print that we were getting wrong. And so one of the first issues that has also almost been used as a synonym for the problems of tractography is the crossing fiber problem. So this was something that was observed very early, that in a voxel where you had just coherent fibers, the orange fibers that you see here, you collect a bunch of measurements from the diffusion weighted imaging scan that tell you that there's high diffusivity in these directions and lower diffusivity in these other directions. And if you fit a Gaussian, which is what the diffusion tensor was, to this diffusivity pattern, then the principal axis of this tensor is going to be well aligned with the fibers in this voxel. And so the early tractography algorithms were simply using those principal axes of the tensors, and following that through this voxel will allow you to reconstruct this fiber bundle successfully. However, in a voxel where you had, let's say, two fiber bundles that were crossing, the orange one and the blue one, the diffusivity pattern that you get from the diffusion weighted imaging scan is still informative. So there's still high diffusivity in these directions, parallel to the orange bundle, and in these other directions, parallel to the blue bundle. But if you try to fit a Gaussian to that, it's not going to be very informative because it's not going to have a clear and meaningful principal axis. So then those early tractography methods just had to stop because they wouldn't know which way to turn. And this set off an effort in the community to come up with better ways to be able to resolve multiple principal orientations within each voxel, what would be called an orientation distribution function or different variants of that. And there have been tons of methods that have been developed since then, and these methods are really refined to the point where we've gotten very good at resolving crossing fibers. So there has been some impression, though, that solving the crossing fiber problem is synonymous with solving the problems of tractography. And so why does tractography still go wrong, even though we've gotten so much better at resolving crossing fibers? And even though in state-of-the-art acquisitions, we get much improved signal-to-noise ratio and resolution compared to what we were getting 15 years ago. So because of those improvements, we can get very accurate estimates of those diffusion orientations in our orientation distribution function. However, the spatial resolution is still limited to, in vivo, at best, about 1.5 millimeter. And there's always going to be more than one axon population in a 1.5 millimeter cube of tissue. And we are unable to resolve certain types of microstructure, even with a very accurate estimate of diffusion orientations. So even if we have a very accurate ODF, in all three of these, for all three of these fiber configurations, we'll get a very similar ODF. And the ODF will tell us that there's a lot of diffusion this way and a lot of diffusion that way. But a tractography method that reaches this voxel will still have to make a choice about which way to turn. And that right now has to be done arbitrarily. And so the typical choice is to go as straight as possible, which basically corresponds to the crossing fiber. So the methods have been designed to be as good as possible at resolving crossing fibers, but then we have other configurations like turning or fanning, where going as straight as possible would not be the correct answer. And so that's why even with great data quality, high SNR, high resolution, we still get some errors. And so what this looks like is when the tractography method takes a wrong turn because of that arbitrary decision that it has to make, it might merge different pieces of different pathways. Like, for example, here, the algorithm was following the corticospinal tract and then jumped onto the corpus callosum and crossed over to the other side and jumped back onto the CST on the other side. So this is something that's obviously wrong, but a lot of the errors are not that obvious to tell apart from the real connections. So because of that, we can still have anatomically meaningless connections, even with very high quality data. And this is not a problem that's easy to solve with current methods, but we have ways to control it by via different thresholds in the methods. So the most typical one is a threshold on the bending angle, where as you relax that threshold, you allow sharper turns, you get more stuff. So you will get more true connections, but also more false connections. So there's always going to be a trade-off between true positives and false positives. And so in an attempt to quantify this, in collaboration with Suzanne Haber, we collected high-resolution diffusion MRI data on post-mortem macaque brains that had received tracer injections in her lab. And so this allowed us to do a voxel by voxel comparison in the same brain of diffusion tractography and tracer injections. And allowed us to trace those ROC curves that show us the trade-off between true and false positives. And so what we found is that in the default thresholds that are used for both deterministic and probabilistic tractography, and the thresholds that would typically be used when you run tractography in the whole brain and try to get a whole brain network, that we're operating at a pretty low true positive, low false positive operating point. So it's a pretty conservative threshold. So we'll miss a lot of the real connections. And in an attempt to compare different methods and different pipelines, this shows a comparison between a bunch of probabilistic and a bunch of deterministic methods in that same brain. And interestingly, we found that in those conservative thresholds that are the default thresholds used by most toolboxes, both of those approaches, the probabilistic and the deterministic, are performing at a similar level. But as we relax those thresholds to get more of the true connections, then the probabilistic methods were better at getting a higher true positive rate at the same false positive rate compared to deterministic methods. And this is something that has been confirmed by a couple of other studies using similar approaches. So then we made those data sets available to the community as part of the Iron Track Challenge, which was an open tractography challenge that we organized in 2 rounds, most of it during COVID lockdown. So most of it was very virtual. So the different teams optimized their tractography pipelines for one injection site, and then applied these optimized pipelines to another data set that had a tracer injection in a different site. And so we saw how consistent those optimal parameters were when we looked at different seed points in the brain. So the outcomes were we recommended best practices in terms of how to analyze the diffusion data. And then we also looked at identifying common failure modes, where most of those different pipelines consistently made errors. So what were those parts of the brain? And interestingly, we found, and the video works perfect. So we found that those common failure modes happen in parts of the brain that involve fiber configurations other than the crossing. So we've gotten very good at resolving crossing fibers, but all these other patterns like branching, fanning, turning, etc., we have neglected. And so that's why we still get errors, and we have to look into that as we develop the next generation of tractography methods. So just before I end, just the outlook of how are these methods evolving, and how much more are we going to learn from diffusion MRI techniques about brain connectivity in the coming years. So one big area of research in terms of diffusion acquisition methods is methods that allow us higher spatial resolution. And particularly trying to go to the mesoscales, quote unquote, so sub-millimeter resolution in vivo with diffusion MRI. And this was a study done by Chiara Maffei that she published at ISMRM a couple of years ago, showing an example from one of the Human Connectome Project data sets, one of the subjects that were scanned on the Connectome scanner at MGH at 1.5 millimeter resolution. And another subject that was scanned with a newer technique that allows sub-millimeter 0.76 isotropic millimeter resolution in vivo. And here Chiara attempted to reconstruct certain small subcortical bundles, so going back to the question of the subcortex and when we're going to stop neglecting it. And the fornix is the big bundle that's shown here for reference and can be reconstructed pretty successfully at both resolutions. But some of those tinier bundles, like the stria medullaris, mammillothalamic tract, fasciculus retroflexus, and mammulotegmental tract, can only be resolved at this higher resolution. The issue is that this scan was actually collected by scanning the same person 9 times, 2 hours each time. So not exactly clinically feasible at this moment, but hopefully with some machine learning techniques in combination with these acquisition methods and the SNR of these acquisition methods improving will allow us to get to the point where this kind of acquisition is feasible in vivo, even in patients. And this is critical for being able to target some of those neuromodulation techniques that were mentioned earlier. And something that I alluded to earlier in the talk is that we have been thinking in a very simplistic way about these large highways of the brain. And so this is another more recent study by Chiara. She has a talk on this at OHBM. So make sure to go to that if you are in Seoul later this summer. It shows that as we go to those higher resolutions here, using ex vivo brains, where it's possible to go to even higher than 0.7 millimeter resolution. So going to 250 micron resolution in small blocks that we then assemble back together within the context of the whole human hemisphere, as well as going down to 3 micron with optical imaging in those same blocks, we see that the superior longitudinal fasciculus, which we typically think as an end-to-end frontoparietal connection, as we go to those high resolutions, it actually turns into a series of relay fibers that connects pairs of regions along the way. And we don't expect that this is going to be the case for all pathways. So some of those large pathways like the uncinate fasciculus, et cetera, we do expect that they'll have some end-to-end connections. But it's important to understand that when we think of these as just a simplified highway, we're losing a lot of that context about what really is connected to what. And so we hope that in the next years, we're going to be able to scale up the techniques that will allow us to do this kind of imaging in ex vivo brains and get to the point, maybe in the next 5 years, if we're successful, to be able to image whole human brains at sub-micron resolution across scales from the sub-millimeter to sub-micron. And so this is something that we're attempting as part of the Center for Large-Scale Imaging of Neural Circuits, or LINC, which is a comprehensive center, a consortium of 8 institutions that has been recently funded as of last September as part of the Brain Connects program, which is an initiative to produce wiring diagrams of the brain at for different species at unprecedented resolutions. So we're specifically interested in our center to image cortico-subcortical projections that are critical for deep brain stimulation to the subthalamic nucleus, both in macaques, where we have the classical tracer injections as the ground truth to validate all the new microscopy and diffusion MRI techniques, and then taking those novel techniques to the human brain to allow us, hopefully, to get to the precision that so far has only been possible with tracer injections in non-human primates, but be able to do that in postmortem human brains and get those ground truth connections that will then allow us to improve our in vivo techniques. And I will end here thanking our funding sources and also putting a little plug and a little update on our new journal, Imaging Neuroscience. So the journal formerly known as NeuroImage, same people, different name, different publisher, importantly, nonprofit publisher. It's been going great thanks to the support of the community. And we hope that in just another year, we'll have a great impact factor. So thanks again to the support of the community. And I will end here and happy to take questions.
Cool. Well, thanks, Anastasia. That's impressive, particularly if you think that diffusion MRI so far gave you the major tracts, and then at the end you show us that's all U fibers. So that's
well, not all, you know.
but at least it would make sense. I mean, these U fibers are so densely present there, no? So yeah, okay. So interesting stuff. Congratulations to this funding initiative.
Thank you.
I think it's really important, and to the journal. Yeah, yeah, good to see that too. So I had one raising hand here already, and then Simon, you can already pick if you find something interesting.
Yep, there is a question here. Will link cover other brain areas?
Wait, I said after our question here.
Come on.
Okay.
Okay, don't fight now.
A brief one here, and then you get to hand to you over, because then you'll introduce our next speakers.
Anastasia, it sounds as if diffusion MRI didn't have a problem with inconsistent terminology. Is this true?
Well, actually, no. I mean, a lot of the terminology comes from anatomy, from classical studies. And there's also, you know, divergent ways of defining the same thing. So even just, so to build that atlas of the main tracks that I showed in one of the earlier slides, that took a lot of, you know, integrating things from the literature and try to reconcile, you know, different definitions. So yeah, maybe we make it look, you know, simpler than it actually is, and maybe that's, you know, to the detriment of the field sometimes.
Okay, Simon, you get one shot now, and then we have another one here.
I have two. Let me pick the one that is a little bit closer to what I was about to ask. How meaningful is structural connectivity given the current diffusion acquisition resolution, all the problems? And I will extend that. Will it ever be useful to study inter-individual differences given the amount of scan time you really need to get to this still not perfect resolution?
Yeah, so, well, my hope is that yes, we will get to that point. So that acquisition that I showed with that was like the 18-hour acquisition, that was already a few years ago, and already new developments to these sequences have been introduced. And I think that in combination with some machine learning techniques to be able to, you know, upsample acquisitions that are sparser acquisitions will hopefully get us there. I do think that is what you raised about the individual differences is very important. Something that we don't always think about is that the errors of tractography are not necessarily, they're not always errors that come from variance, from noise. Some of them are errors of bias. So I showed those the areas where many tractography methods make errors consistently. So those errors will be there consistently across multiple brains and even multiple species, those, you know, tricky turning points, etc. And so even if you average, you know, data from 1,000 brains, sometimes we think, oh, I'm going to average data from 1,000 brains, I'm going to solve some of those, I'm going to get rid of those noisy fibers. But those errors that occur consistently across brains are not going to be averaged out, even if you average 1,000 brains, because they're biased, they're not variance. And so that's why it's so important to really push those methods for getting the higher accuracy in individual scans and in individual patients rather than relying on averaging.
Okay, and there's a question with a yes-no answer. Will link cover other areas, but cortical subthalamic?
Sorry, what was the question?
Will the link actually focus on cortical subthalamic, or will you also look at other networks and areas?
So the goal is to image that entire block of the brain that contains the projections from the motor, premotor and prefrontal cortex to the subthalamic nucleus. And so those connections are there are use case but the data will be there and will be publicly available for others to mine, to study other connections. And hopefully, once we show, fingers crossed, the feasibility of scaling up these acquisition and analysis techniques, then the idea is that in the future they'll be applied to entire brains.
Thanks a lot.
Thank you.
Not
A
quite
lot of
yet,
work
no,
to do.
but thanks a lot for the great talk, and we're really looking forward to the output
Thank
of the
you.
link. And
Thank you.
I have one more question here from Martin, and then we hand over to Simon again, who will introduce Yong-Hee, who has been briefly on. Now he is there. Great. So one more question here, and then you can prepare for the online setup.
Yeah, thanks for the talk. So I know some neuroanatomists like Henry Kennedy that are quite skeptical on the diffuse and fMRI because the correlations when you correct for distance are quite weak, in macaques. So that was 2016, I think, done on you. So did it get any better, or what's your take on the relation to anatomy?
Yeah, so the goal of those validation studies was to exactly try to quantify that. And indeed, the further you have to go, the more opportunities there will be to go through those tricky parts where, things get messed up consistently. So our goal was not to decide if we should be skeptical or not. It was more concretely to decide what are the next steps that we have to take to improve these methods. So try to pinpoint exactly what goes wrong so that then we can think about the next generation of methods and how to fix that. So yeah, I would say that we're hoping to do more than just be skeptical about it. Of course, being skeptical is the first step towards doing those validation studies and justifying them to funders, etc. So it is important to have that skepticism, but it's not an end point, right? The skepticism. So already it is much better.
In the interest of time, may I ask you to continue that discussion?
Okay, in the beer garden.
Okay, you have 25 minutes for a talk, Simon, and then 5 minutes for discussion moderation.
Okay, so Yong-Hee, get ready. There you are. Welcome. And while you are hopefully in 2 seconds setting up your talk, I will take 30 seconds to introduce you. So it's my pleasure to introduce Yong-Hee. He is a professor of the State Key Laboratory of Cognitive Neuroscience and Learning, as well as a principal investigator of the IDG McGovern Institute of Brain Research. Both of that is at Beijing Normal University, and there his lab uses multimodal neuroimaging and computational modeling to investigate, and this is, I think, right at the heart of what the symposium is about, to investigate how the human brain connectome is organized and topologically disorganized in health and disease. And I covered the time until you have your slides ready. Yong-Hee, the floor is yours.
So actually, it's great to see you. So thanks, Simon, for the kind introduction, and thanks for the kind invitation from the committee. So it's my great honor to have this great opportunity to talk about the summary of structural MRI-based connectivity. So actually, for structural MRI images, we know the MRI signals varies across different tissue types, such as gray matter, white matter, and CSF. But we know the gray matter contains small cell bodies such as neurons and glial cells. But white matter is mainly composed of long-range nerve fibers along with supporting glial cells. So structural MRI provides very important neuroanatomical information to qualitatively and quantitatively describe the shape, size, and integrity of gray matter and white matter structures. So actually, there's many morphological features of the brain which can be estimated from structural MRI data. So for these morphological features include gray matter volume, cortical thickness, surface area, circle depth, and the fractional dimension, and so on. So I'm not going to much details about each morphological measure, so just give 2 examples. So the first one is cortical thickness. So we know the cortical thickness measures the distance between the gray-white interface and the gray CSF interface. So cortical thickness reflects the arrangement, size, and density of neurons, neuroglia, and nerve fibers. The surface area, it described as the area of the exposed cortical path surface and hidden area of the cortex within cycle. So surface area is thought to be driven by the number of cortical columns. So the different morphological features carry different morphological properties and have different neurobiological bases. So these morphological features can be extracted by using different methods, including manual tracing method and automatic analysis methods, such as VBM, DBM, and SBM. So in this figure, especially panel A showed some shared pre-processing and processing steps among these methods, including non-uniformity correction, tissue segmentation, and registration to standardized anatomical template. For SBM analysis, so we need more surface reconstruction and surface registration. So using this method, so we can obtain morphological features such as grammatic volume, density, deformation field, cortical thickness, and so on. So in the past years, so many studies have used this method to study grammatic morphology. Especially, they can look into grammatic loss in brain disorders. But the regional morphology is not isolated, but correlated between different areas. So there's two main methods for examining interregional morphological association. So one is population-based. The other one is individual-based. For population-based methods, so the basic idea is there are remarkable individual differences in the structure of brain regions. So just give example. So we can see the two areas, the left hippocampus and the right hippocampus. So if we measure the volume of hippocampus across individual, here we can see the very strong correlation between left and right hippocampus across individuals. In other words, if one individual has a smaller volume in left hippocampus, so these individuals has also smaller volume in right hippocampus. So for interregional morphological association based on population method, so they need to estimate the morphological correlation by using structural MRI data of a group of participants. So we cannot generate individual morphological brain connectivity using a single MRI scan. So just give several examples about population-based method. So the first one is about grammatic volumetric correlation based on postmortem data. So from this figure, we can see there's strong correlation of grammatic volumes among different areas in the viral-related system. So it's another study done by Woodruff by using structural MRI data. Actually, the first speaker have mentioned this reference. So they measure grammatic volume in healthy controls and patients with schizophrenia. And we can see in healthy controls, there's stronger volumetric correlation between two frontal and temporal areas. But for schizophrenia, so this correlation was reduced. And also some seed-based connectivity analysis. So in this study done by Andrew McKelly, so they pick up a lot of seed regions and calculated the correlation between the seed area and the rest of the brain. So we can see very strong correlation between homologous regions here. So it's another seed-based correlation analysis done by Jason Lurch in 2006. So they pick up seed region in Broadband 44 area and do the correlation with the rest of the brain. So we can see this very strong correlation between Broadband 44 and Wernicke area. So this pattern is remarkably similar to language-related white matter pathway. So besides pairwise correlations, so we built morphological brain networks using cortical thickness correlations. So in this work, we demonstrated that many strong correlations indicated by the circles is homologous regions. And also we found some short-range connections and a few long-range correlations. So these connections are very important to link different modules. So in this work showed the first time the human brain networks had robust model properties with short-range correlations and long-range connections, as well as module organization. So actually the morphological brain networks have been extended into the use of other morphological features, such as grammatic volume by Daniel Bassett and the cortical surface area done by Sandberg Diaz. So in this work, they compared topological organization between surface-based network and cortical thickness-based network. So they found very significant difference in many topological parameters. So we can see here. So actually, it's not surprising because we know that different morphological features carry different morphological properties, and they have different biological bases. So this is why these different morphological networks have different topological organization. So for population-based method we just mentioned, so we cannot use population-based method to build individual brain networks using single structural MR data. So actually in recent years, there's many studies have developed individual-based method to extract morphological brain networks using a single structural MR scan. So actually this individual-based method can be classified into low order and high order. So for low order method, it describes pairwise relations between different areas in local morphological features. So this kind of low order methods can be categorized into 4 classes. It's correlation-based, divergence-based, distance-based, and division-based. So for correlation-based method, so it's widely used and very simple for quantifying the linear relationship between different areas using single feature or multi-features. For divergence-based method, it involves estimating a probability density function from morphological features within a region of interest. For distance-based method, it uses many distance functions to quantify the dissimilarity between 2 different areas, such as one-dimensional Euclidean distance and multi-dimensional Euclidean distance. For division-based method, so it estimates the similarity between different areas based on division of morphological features from a reference distribution. So if we use division-based method, we need to build normative growth model to find some division information. So for high order method, it describes the interplay of low order morphological relations between different brain regions. So if we want to use high order method, we need to first build low order methodological relations. So after that, we can use Pearson correlation and the multi-layer model to examine interregional relationship of low order morphological correlations. So just give several examples about individual-based method. So the first paper done by Betty Chijin in 2012. So in this work, so they use structural MR data to obtain the 2 cubes. For each cube, they have grammatic morphological information of its neighbors, and they do correlation in morphological features between any pairs of cubes, generating the individual morphological brain network. So if we look at the highly connected degree in individual brain networks, so we can see, so these regions, highly connected regions, are mainly located at lateral temporal and medial frontal and parietal regions. So and also there's very strong correlation of the node features between the 2 different scans. So this is another individual-based method developed by Ed Boomer's group. So they developed a new technique for network mapping based on interregional morphological similarity of using multiple parameters, measure use the structure MR and the diffusion MR data. For structure MR data, so they can obtain several different morphological features, such as green matter volume, surface area, cortical thickness, and mean curvature. Now for diffusion imaging data, so they can estimate several myelination indexes, such as fractal anisotropy, mean diffusivity. So actually, there are 10 morphological features in total. So they can do the correlation of these features between two different areas, the resulting interregional morphological similarity network they call MSN. If we look at the network hubs in the MSN network, so the highly connected nodes are mainly located at lateral temporal and frontal parietal regions, as well as medial parietal and frontal areas. And also they obtain the model organization in the MSN network. So if we look at the model organization, including anterior frontal area, posterior occipital area, and the lateral temporal area. So this model organization is mainly anatomically constrained, so which is very similar to diffusion structural connective networks, but it's very different from functional connectivity modules. So it's another method also developed by the same group. So they proposed a morphometric inverse divergence to estimate morphological similarity between different areas based on divergence measure between their multivariate distribution of multiple MR features. So the basic idea is they generate cortical surface based on the mesh reconstruction using structural MR data. So if we pick up the two regions of interest, so for each region of interest, there are many vertices within this area. For each vertex, they can estimate 5 morphological features, including cortical thickness, volume, surface area, mean curvature, and socket depth. And they can use KL divergence to estimate the similarity between the two areas in terms of their multivariate distributions. So they have compared minor network and MSN networks. So let's say the minor network showed the high resilience to noise features and a robust to different postulations and have more interhemispheric connections and have more consistent with cortical occipital architecture indicated by green colors as compared to MSN network indicated by purple colors. So it's a very nice method for single morphological brain network. So the next question is, what's the relationship between morphological brain networks and structural connectivity networks? So about 10 years ago, my colleague Gao Langgong have used structural MR data and diffusion MR data to build morphological brain networks and structural connectivity networks in a same group of participants. For thickness network, so we have positive thickness correlations and some negative correlations. But for positive thickness correlations, we can see about 60% show the convergence with the diffusion structural connectivity indicated by red colors. If we look at brain hubs between the two brain networks, for precuneus is the common hubs, but there's still very different patterns in brain hub region distribution between two networks. So if we do correlation between morphological brain networks and the diffusion connective networks in terms of regional efficiency, we can see very strong correlation with a correlation coefficient of 0.4. So what about morphological brain networks and structural connectivity networks in monkey? So in the two papers, they compared individual-based morphological networks, MSN and MIND, and track tracing external connectivity in monkeys. For MSN network, so they found strong correlation between group MSN edge width and track tracing structural connectivity edge width. So the correlation coefficient is about 0.34. So let's look at MIND network. So morphological similarities estimated by MIND were generally more strongly correlated with track tracing width, regardless of different populations and feature subsets as compared to MSN metric similarities. So from these results, we can see the MIND network is much more closer to track tracing external structural connectivity in monkeys. So there's very close relationship between morphological brain networks and diffusion structural connectivity networks, or track tracing connectivity network. So the next question is, if structural connectivity patterns shape the maturation of coordinated cortical morphology during development. So we address this issue. So in this work, we showed cortical singing patterns from between childhood and adolescence. So we found cortical singing patterns which are mainly focused on frontal and lateral parietal regions. And also we found very strong correlation between node cortical maturation and structurally connected defined neighbors. So in other words, so if one region showed cortical thinning during development, so its structurally connected neighbors also shows cortical thinning. So it's synchronized cortical thinning patterns in the context of structural connectivity. So the next question is, what mechanisms behind these structural connectivity constraints on cortical morphology maturation? So we propose a network-based diffuse model for predicting cortical thickness maturation. So the basic idea is we have different neighboring scale. For each scale, we can generate diffuse probability matrix according to its neighbors. And for each scale, we have diffuse probability matrix. We can use this matrix to predict cortical thickness maturation. So the predictions are very significant. So from these results, we clearly see, so the cortical morphological maturation can be shaped by structural connectivity patterns. So what about the relationship between morphological brain networks and functional connectivity networks derived from rest fMRI? So it's a very beautiful study done by William Seeley in 2009. So they use rest fMRI data and structural MRI data in group of participants based on seed-based connectivity analysis. So we can see that very strong correspondence between the two different networks. But if you look at the gradient architecture of the brain networks, so they're very different. So for MSN network, so the first gradient is from primary motor cortex to visual cortex. But for functional gradient, the first architecture is from primary sensory motor and visual regions to associated cortex. So the very different gradient architecture. But from these results, we can assume that so within each system, possibly there's some good correspondence between morphological brain networks and functional connectivity networks. But the whole network architecture, they're very different, possibly due to some long-range connections difference between two different modalities. So what about robustness, reproducibility, and reliability of morphological brain networks? So they are very important for the brain connectivity studies. So for robustness, so this measure quantify how much consistent in results when the same evaluation is performed on the same participants with different analysis method or parameters. For reproducibility, so it quantifies how much consistent in results when the same evaluation is performed to different participants or independent datasets. For reliability, it quantifies how much consistent in results when the same evaluation is performed to the same participants across different time. So actually, the several studies have validated this performance of morphological brain networks. So generally, morphological brain networks consistently show high robustness, reproducibility, and reliability, regardless of the specific method used. But the performance is different over the different methods. So given the importance of high robustness, reproducibility, and reliability of morphological brain networks, so actually in the past years, there's many studies have used morphological brain network analysis idea to study normal development, aging, and brain disorders. But in the future, it's very important to establish lifespan morphological brain network curves to serve as a baseline reference to detect individual difference, identify biological subtypes, and localize potential neuromodulation targets for brain disorders. So finally, actually, we want to clarify. So it's the regional morphological association, the type of brain connectivity. So actually, in this talk, in the literature, there's many different terms, such as morphological correlations, structural correlations, structural covariance, morphological connectivity, similarity, and so on. So let's recall the definitions of functional connectivity and structural connectivity. So the functional connectivity describes the typical dependencies among remote neurophysiological events. So structural connectivity describes anatomical connections linking a set of neural elements. So in this review paper, so we tried to define the morphological connectivity as the statistical interdependence of local morphological features between different areas. So just give a quick summary. So in this talk, we can see structural MRI can be used to study the regional morphological connectivity of the brain. So especially morphological connectivity networks can be constructed by using different methods, such as population-based method and individual-based method. But for individual-based method, it's very important to build individual brain networks using the single structural MRI data. So we know the structural MRI is very easy to collect. So it's just take about 5 minutes. So the morphological connectivity networks show critical network properties, such as small module, gradient, and halves, just like functional and structural connectivity networks. But we have noticed that morphological connectivity networks only partly reflect the structural and functional connectivity. So they have very different patterns. So morphological connectivity networks shows high robustness, reproducibility, and reliability, so which is very important for the application studies. And also morphological connectivity networks show the potential to detect individual variability and identify subtypes in normal development, aging, and brain disorders, especially for young children and some specific patients with neurological and psychiatric disorders. So finally, the neurobiological basis behind morphological connectivity networks need to be elucidated. So in the future, animal brain imaging studies would be very important to bridge the links between molecular and cellular properties and microscopic morphological connectivity networks. So finally, I would like to thank my collaborators, Alan Evans from McGill University. So actually, I did my postdoc about 20 years ago. I started my work at Alan's lab, especially morphometric connectivity networks. And also, I think John Chen from McGill University, my colleague Gao from Beijing Normal University, and my former students at Jinghui Wang, who is a professor from Guangzhou. So thank you for your attention.
Thanks a lot. Super exciting talk, super interesting findings, and you stayed well on time, so perfect.
Thank you.
see any questions in the chat yet. So, Martin, are there raised hands?
Yeah, we would have two here. So shall we start?
Please go ahead.
Okay, you were first, and then.
Yeah, thank you. I really enjoyed your talk, and I really like, similar to radiomic similarity networks, I like this way of looking at the structural data also. I just wondered, apart from the open question of the neurobiological meaning, do you find that this has predictive utility that's really unique and possibly better than just very average structural measures that are first order, like thickness, you know, volume, even also morphological features like shape?
Yeah, I think it's a very important question. So actually, for the neurobiological basis of individual-based morphological networks, actually the several studies have utilized gene expression profiles to explain a molecular basis of the networks. So, but we have to notice that they have very different subjects, and for gene expression data, there's only a few subjects. It's also this is a remarkable individual difference. Although that they have give some preliminary linkage between morphological brain connectivity and macrostructural events. So if we want to look at the more details, especially the direct linkage between macroscopic brain connectivity and neurobiological properties, I just mentioned possibly the animal brain imaging would be very important direction, because we can modulate the animal brain. We can provide more direct evidence to link macro scale and macro scale properties.
Okay, thank you. Another question. The question I have is about the definitions. This is a nomenclature meeting. And in particular, I'm confused by the definition of morphological connectivity, which instead to me is a similarity between regions. So why we're confusing similarity with connectivity? So why should we not call it morphological similarity and leave the connectivity out? That's a provocative question, of course. Would like to hear your opinion.
Yeah. So actually, we follow the definitions of functional connectivity. So for functional connectivity, just statistical interdependence between different areas. Actually, the several speakers have talked about the definition of functional connectivity. So we can use PET studies, PET data to look at interregional functional connectivity across individuals. Actually, the previous PET studies have looked at this. So the idea is very similar to morphological correlation across subjects. But for morphological brain connectivity, also we can use individual-based method to build a morphological brain networks from a single structural MR scan. So the connectivity means in the morphological domain, we just look at the morphological similarity between different areas. So actually, again, that's the neurobiological basis of the morphological connectivity needed to further studies, and also what specific and common properties among morphological connectivity, functional connectivity, and structural connectivity. And also, it's very important to find out the specificity and the similarities among different modalities. So actually, I welcome any discussions about this connectivity. It's very specific connectivity definitions.
Yeah, thank you, Young. I think that's definitely a question that we're gonna have to address later on. Also, Simon, you want to have a brief comment on that?
Yeah. I frame that comment as a question. Is anything that is correlated connectivity? Because Young, I feel you're getting right at the heart of what this meeting will struggle on. We've heard people having very mechanistic ideas of connectivity, but if you look at resting state fMRI, if you look at PET, it's really in the same boat as the structural connectivity you mentioned here, where we call anything that correlates connectivity. So should we actually split this into connectivity being clearly mechanistically defined and correlation, or find another good word here that is telling something about the relationship between brain regions without giving a suggestion of actual mechanistics?
Yeah, I think, Simon, that was a bit what Ariana also presented in the beginning. No, like we had the covariance for these non-biologically linked signals that just fluctuate, and connectivity originally like physiological covariance of spiking activity, where you assume some neurobiological signaling.
And I think the talk by Young really illustrated that so well, and in particular the discussion, I think, put the finger right into it. Can we get away with using one term, i.e. connectivity, for both of that?
Yeah, thank you. Actually, I know that the topic of this session is what is brain connectivity. I welcome any feedbacks and suggestions about this topic. Yeah.
Thank you, Young. Thank you, Young, for joining and giving this inspiring talk here that we could all follow here in Munich and online. That was a great presentation. And Simon, thank you for moderating that now. I would give Young another hand of applause here from Munich.
Thank you very much. Thank you.
And Simon, I'm going to introduce now the final speaker of this session. Igor is your colleague. Yeah, okay, you will set up the presentation in the meantime. So the final talk now, before we go into a lunch break, and I'll give you a brief, we hear more details about how it's continue afterwards. So the next speaker, the final speaker of this first session now, this public session, is Igor Yakushev. Igor is head of neuroimaging and a senior consultant in nuclear medicine department here at the Technical University in Munich. And he leads the research group Multimodal Imaging of Normal and Pathological Cognition. And the group's effort is to understand the mechanism of normal and pathological cognition, relying on the concept of brain connectivity and using and merging imaging methods such as PET, structural and functional MRI, and diffusion MRI. He's co-founder also of this molecular connectivity working group that has been organized today. And yeah, there he is coming. So welcome, Igor, and looking forward to your talk.
Thank you, Valentin. Thank you for the introduction. Thank you for the introduction, and thank you guys, Valentin and Simon, for the great moderation. Yeah, I have no disclosures regarding this talk. Outline. Well, I will first briefly define molecular imaging, as a large portion of the audience has a different background. Then try to justify the use of molecular imaging in the field of brain connectivity. Then we'll show you the ways how to estimate molecular connectivity, disputable term. Let's see how it goes in the afternoon or tomorrow. Molecular connectivity, what can be used for, which biological targets can we use to derive information about brain connectivity, and finally, yeah, the issue of lack of standardized nomenclature. So, molecular imaging. This is a definition. The keywords are visualization, quantification, cellular and subcellular level, and living subjects. How does molecular imaging work actually? On the example of the positron emission tomography, which is the major tool of molecular imaging, everything starts underground in a radiopharmaceutical lab. Yeah, okay, it works also. The first step is production of radionuclides. Radionuclides, we use only radionuclides that decay via positron emission, therefore positron emission tomography. In a lab, a radionuclide is coupled with a molecule of interest, for example, glucose, resulting in a tracer. So why tracer? Because the substance traces a biological process without interfering with it. So we talk about nanomolar range, very low concentration. Then there is a quality control, and if all is good, tracer in the form of a small amount of fluid is injected intravenously to a person. And immediately or after a certain time delay, we start data acquisition, scanning, and as any radionuclide has a certain half-life time, which is of course known, after a certain period of time there is decay, positron is emitted, meets an electron, and it results in a so-called annihilation, emergence of two gamma rays which go in the opposing direction and can be detected by PET detectors. And higher the concentration of the tracer in a brain, in a certain region of the brain, more gamma rays arise, the higher intensity in the image. So let us compare three methods which are used to measure neural activity. EEG, as we learned, is the oldest one. Bolt fMRI is by far the most popular method to measure functional connectivity, and FDG PET, fluorodeoxyglucose PET, so glucose PET, is the most popular method of molecular imaging. So spatial resolution is in centimeter range for EEG, 2 to 5 millimeters Bolt fMRI, and 3 to 8 millimeters for FDG PET. One with star because it refers to recent technology of total body PET CT. So with these scanners we can reach 1 millimeter spatial resolution. Temporal resolution is best definitely for EEG, Bolt fMRI seconds, FDG PET in a conventional case minutes, but I will mention later method of dynamic PET acquisition of functional PET where we can be at the level of seconds. Data mode, dynamic, static, it's a sort of typical mode of data, FDG PET data, but again, there are recent developments in the field where data are acquired and analyzed dynamically. We'll get back to this later. Scan duration is quite similar between the methods. Of course, it's all everything approximately. So 10 minutes, Bolt fMRI, EEG, FDG PET. In a conventional case, in a clinical case, 10 minutes are enough. But if you want to derive this temporal information, so to analyze data dynamically, then you need more time, 60 minutes, 90 minutes, or even more, depending on the tracer and on the process under the study. Radiation exposure, no, yes. Costs very low for EEG, low for Bolt fMRI, and FDG PET high or very high. So, and these two factors really explain why FDG PET or PET in general is much less available than the other two techniques. But it has also advantages. Neural substrates, I hope fMRI people will not strangle me, Lucinas. E.g., well, very direct, we measure neural activity of neurons, electric activity of neurons. Bolt fMRI, well, we think of neurovascular coupling. So Bolt is a sort of surrogate of cerebral blood flow, cerebral blood volume, and cerebral metabolic rate of oxygen. FDG PET, I would say, measures neural activity in a more direct way. What do we measure exactly? We measure activity of hexokinase, the enzyme that phosphorylates or catalyzes the phosphorylation of glucose. Signal-to-noise ratio is higher for FDG PET. Reproducibility is also higher for FDG PET, and quantification is possible with FDG PET. So we can derive a number from each region in the brain, also in the case of clinical scan. And clinical utility is established for EEG, quite limited for Bolt fMRI, at least clinical, so in a patient, and FDG PET an established method in routine clinical settings in the field of neuroimaging. So I think FDG PET at least deserves, based on these characteristics, deserve to be tested, to be applied in the field of brain connectivity. Why molecular imaging? Another component or another argument. FDG PET is of course not everything. PET can much more. And you have seen the slide already from Ariana. FDG PET or PET can measure components of neurotransmission systems, for example, at the presynaptic level, dopamine, serotonin transporters, at the postsynaptic level, new opioid receptors, and D2-like dopamine receptors. And we can also measure pathology, so pathological molecules which do not exist in a healthy subject, but may appear in a patient, for example, tau or amyloid. So molecular connectivity, again, the term that we can criticize afterwards, but so far we have defined it by analogy with fMRI, more or less, molecular imaging of brain connectivity, and this definition has been cited a few times. Functional connectivity is considered as statistical dependencies among remote neurophysiological events, and if we translate it to molecular imaging, we have this definition in red, statistical dependence between regional measures of molecular imaging. So far, so good. How can we estimate molecular connectivity? There are two principally different ways. Intrasubject, so the way that Yonghe also introduced, and intrasubject. This is more similar to the fMRI world. I would start with the intrasubject estimation. Nice slide by Ariana Sala. So it's probably easier to understand, at least for fMRI people. So here we have multiple measurements of tracer uptake over time in one subject. And we assume that two regions are connected if their tracer uptake covary over time, like in this case. Intrasubject is trickier because one time point is one subject. So we measure in the same regions covariance, but not over time, but across subjects. So we have subject series instead of time series. There is no dimension of time. And this is what people also criticize. Also the terminology in this regard. And In this context, I think it is instructive to talk about ergodicity or ask how actually equivalent are patterns of intersubject covariance to patterns of intrasubject covariance. And the question is nothing else as a special case of ergodicity, which is interchangeability between variability across time and across ensembles. This is not, it's a quite old and well discussed phenomenon in the past, also in the field of neuroimaging. And I found a very interesting statement in the short course by Olaf Sporn's Analysis and Function of Large-Scale Brain Networks by Anthony McIntosh. And I share this statement. Some neuroscientists are concerned about which source of variance across tasks or across subjects is best for estimating neural interactions. The issue of which source of variability is correct is not unique to neuroscience. Within subject analysis assesses the direct relation between regions, while across-subject analysis indicates the stability of that relation. These are complementary, not contradictory pieces of information. And in my opinion, this intersubject data may be suitable to characterize population-based or population-shared portion of a pattern, in our case, connectome. Here are results of, from the group of XIND who addressed this issue of ergodicity. ADNI data, Alzheimer's disease neuroimaging database. XIN included 72 subjects who have been scanned at least 5 times, 5 to 9 times, and calculated the overlap between this cross-individual correlation and within individual correlation, and the Spearman correlation coefficient was quite high, 0.77, which, let's say, speaks for the presence of a relevant ergodicity in this case. But of course, it is a degree and not a binary index. I will get back to this in a couple of slides. The second principal way is intra-subject, and this approach becomes increasingly popular. What do we mean? There is a term functional PET by analogy with fMRI, functional MRI. This approach uses a constant infusion technique, so tracer. So far as I know, it has been shown only for FDG. Tracer is continuously infused over time, and researchers measure changes in glucose metabolism in response to a stimulus or at rest in subjects. Here, one example of a visual stimulus with a frame duration of 30 seconds. You see there is a stepward increase in FDG in the brain, in the visual cortex. And these are data at rest. So there are fluctuations of FDG over time, so similar to Bolt fluctuations. And the amplitude is low, and but similar to Bolt fMRI, if we average the signal over subjects, there is a meaningful pattern of correlations between these different regions. So also a reasonable way to estimate brain connectivity. So, and back to ergodicity, I think it's a matter of temporal scale, right? Temporal resolution. Here, if we take the case, a clinical case where we have one image per subject, and this is really the majority of PET data are available as only one image per subject. And if we have a few PET scans with conventional, so low temporal resolution, the ergodicity is quite high. But if we increase the temporal resolution and go in direction intra-subject estimation, this ergodicity would decrease. And I know that Sharna Jamada and Alessandra Bertoldo found that ergodicity decreases, or this overlap between inter-subject and intra-subject correlations decreases. Which statistical methods can be used to estimate molecular connectivity? Basically, more or less the same which I used, for example, in functional MRI. So this is seed-based correlation, where seed can be a region of interest or a voxel. And, for example, independent component analysis data-driven technique, typically applied at the voxel level. Maybe a particular method which is more common in the case of PET is so-called sparse index covariance estimation. Why is it useful in the case of PET? Because it is quite robust to a low number of subjects. So when the number of subjects is lower than the number of regions, which is quite common for PET data, this method may be useful. Now to biological targets. Neural activity is by far the most common biological target in the field for PET. And we found a very, a high, at least spatial overlap between resting state networks from fMRI and PET data. So the same subject was studied simultaneously with fMRI and PET. And inter-subject, right? So, and still we can identify at least some common established resting state networks from this inter-subject variability. So it is not, I think, simply a statistical epiphenomenon, but we have some plausible information from this, we can derive from this inter-subject variability. And one argument in favor of this biological meaningfulness of this inter-subject variability is here. So you see overlap convergence ratio, so basically Dice coefficient for 3 measures of, or 3 estimates of brain connectivity, overlap with structural connectivity from diffusion MRI, right? So blue is fMRI, brown is FDG PET, inter-subject estimation, and yellow is gray matter covariance. Well, this is FDG covariance, this is gray matter covariance in gray matter volume, right? And we see that the overlap is, for FDG PET is much higher than for gray matter covariance, because there is ongoing discussion if this this covariance is a sort of statistical epiphenomenon and doesn't reflect really brain function. But I think when it is closer, FDG PET covariance is closer to functional connectivity and not to gray matter covariance. This speaks in favor of a functional substrate of this covariance. Another common target is neurotransmission. Why is it good? Because major neurotransmission systems are well defined, so the projections are well known. Here an example of a projection from dorsal raphe in the serotonin system and to the ventral striatum. It is a well-established projection. And if we calculate serotonin transporter in the dorsal raphe and in the ventral striatum and calculate a correlation, simple correlation, it is very high. And this correlation is disrupted in patients with major depression. And finally, also proteinopathies data can be used to estimate brain connectivity. Yeah, the case of tau PET. So it is known that tau can transmit transsynaptically, similar to prion diseases. And if we follow this way, this propagation we can derive information about brain connectivity. And in fact, there was high correlation between functional connectivity and tau covariance across subjects independendly of clinical identity. So also in healthy subjects and AD patients and subjects with ischemic brain disease. So, and finally, yeah, what is the problem? The problem is big. It's simply, I think, in our area is a chaos regarding nomenclature. One term is used with different meanings, and other way around, different words may mean the same, more or less. And this is just a few problematic questions. For example, what is metabolic connectivity? It is commonly used in the context of FDG-PET, but metabolism can be also estimated using other tracers, so not only glucose. Should only intra-subject estimation be used or interpreted as index of brain connectivity, or also this inter-subject can be used? Biological targets, for example, is amyloid PET really reflective of brain connectivity or only tau PET? Because the question of region by region transition of amyloid has not been proven. Spatial scales also not trivial, while results of ICA are commonly interpreted as resting state networks, right? But PCA results, which show quite large clusters, which do not really, or can be hardly assigned to anatomical regions, are they reflective of brain connectivity? So these are just a few questions that will be discussed later on today and tomorrow. And finally, yeah, the major challenge is to differentiate between real connection and a spurious one. And yeah, we have much to do. Thank you for your attention.
Nice, thanks Igor, also to giving us this introduction to PET methodology first of all, which is definitely not common anymore that much, but maybe rising again compared to the beginning of brain imaging. And yeah, and I found it still striking that we have this high or this consistent continuation of within subject coherence to between subject. And so what is the underlying similarity between the measures that we find it consistently within a subject and then even across subjects? I hope that it's something meaningful. Simon, you also joined, I see. Great, for the final discussion. Do you have already questions online?
Not yet, but I can just invite everybody again to post questions online.
Because in here is, yeah, okay, I have, now you have to, Matthias. Or you wanted to comment something? No. I have a question. Yeah, great.
That was the same provocative question, Matthias.
It's a simpler question than before. So you said that PET is one of the few, neuroimaging modality as a clinical applications. While MRI, especially the functional modality, do not have clinical applications. Connectivity does not have clinical applications. Okay, it's not like by bringing PET to connectivity, we are shooting ourselves on the feet and getting far from the real world. So in other ways, should we have should we put before the questions that we want to resolve with the connectivity, rather than just doing connectivity and try to find questions that we can solve? That was the question I have for you.
Thank you. Well, I respectfully disagree. PET is already used for clinical, or let's say PET connectivity, but not strictly connectivity, but the PCA analysis, and Chris Harbeck and other people have shown that this multivariate analysis, these patterns better differentiate between, for example, dementia disorders, patients with dementia disorders, than univariate patterns. Yeah, if it is really, brain connectivity is open question. But I would say also provocatively, do we want a clinical application and then the mechanism is secondary? Or we want to understand the mechanism and then, not focus too much on the clinical application. And I tend to go, at least in the context of this event, for the second direction. Yeah.
Thank you. There's one in the back. Simon, you're still collecting, or?
I'm still monitoring the chat, but
Yes.
nobody from the online side has one. I have one question, but go with the one in the back first.
Thank you.
Thanks, Igor. Regarding what you just said, I actually have a question. As you know, I'm not a clinician. I don't work in the clinics. So the only insight I have regarding clinical applications come from you and other collaborators. Regarding what you just said, as far as I understood, in the clinic, sometimes you don't focus so much on the mechanisms of things, right? So as at least most of my colleagues working in neurology, they say, well, we try a medication, and if that medication works, it can be for another problem, then we go like, okay, this is good. And I think we have the same question with, or the same issue with connectivity, right? We don't know exactly what it means, but we know it works. We know it works when it comes to distinguishing different stages of disease, detecting early signs of disease, and that it correlates also with clinical measures. So I don't know, do you still go for the second approach, taking this into account? Because this is a struggle I have sometimes with, convincing people regarding how valuable are the things that we are doing, right? They ask me, oh, we don't understand it. I was like, you don't understand lots of things, and you use them, so why?
Well, the ideal situation is, of course, a combination that we help the patient and understand why. Yeah. And this is the ultimate goal, and at least in academic settings. We can start, or let's say we can try to estimate brain connectivity using PET, and then go to the individual level, like Yonghe showed. It's basically with a clinically available data with just one image per subject. It can be easily translated to PET. Or we apply this principal component analysis without trying to understand if the output has any functional meaning or any anatomical pattern, meaningful anatomical pattern. Yeah, that's my answer.
Simon.
Yeah, maybe I can
as a wrap-up question, basically. So
yes.
It has to be a good one.
Okay, maybe it's a primer for the future discussions. So we already talked about maybe we need two terms in terms of two different kinds of connectivity that needs to distinguish, that is causal, mechanistic, that is correlative, but maybe we also need to distinguish two different goals. It's to either understand the brain, and this is where, for example, PET connectivity can very strongly contribute, whereas the opposite aim would be single subject biomarkers. So potentially we should splice up or split up the field of connectivity imaging into these four quadrants, and maybe that helps us to resolve some of the confusion, which is, I think, not just nomenclature.
No comment, Simon. Fully agree.
We have Lucina. Okay, do you want to take Igor's mic? And then So
No, just a small comment. I think we already saw on a slide from our earlier speaker that we have already decided there's some divisions, right? Functional connectivity, effective connectivity, structural connectivity, and these different methods we talked about this morning give us insight into each of these definitions, right? Functional connectivity is maybe just correlation, right? Or structural, maybe more we can get at with diffusion and anatomical tracers. So I think we had already, if not consensus, we have some definitions there, different ways of thinking about connectivity. They don't always map one to one to one, right? Because of course there are different definitions of connectivity. So I don't think we're completely lost as far as the definitions go. But as Simon mentioned, we can get even more refined in our definitions of those three types of connectivity, where we think they're most applicable, for example, or where we think they're better to look at in the intersubject way versus the intra-subject way. I think we're not as far away from consensus as maybe it's not so hopeless maybe as we started to think.
Yeah, I think there might be even some help in unifying these methods, no? Because as we heard now really nicely, I think that's the profit from this conference, from this workshop so far, that you capture different aspects, no? And we had this discussion in the beginning about what does an area signal really implicate, and then you have maybe diffusion that shows more the connection between those, but you have locally completely different things going on in terms of hemodynamic responses versus then PET tracer amplitudes and so on. So therefore, by unifying that, maybe it will help to gain even more about what connectivity could mean, instead of defining the terms without knowing what the real measure is below that. I guess, yeah, we will close here. We are basically almost in time. That's nice. And I briefly hand over to Sylvia, I guess, to wrap up and give some insights about next steps. And I just lost Simon, you're still here. So also thanks to you, Simon, for joining online and helping to moderate that. It was a real pleasure with you here.
Thank you. So before closing, we would like to thank Munich for hosting us, and in particular to the University Hospital of Munich for hosting us for this part of the symposium, and also for the closed part that will follow. And we would like to thank supporter fundings such as DFG, Bruker, Curium, and PMOD that economically supported the realization of this symposium. And yeah, this is the end of this open part to the public. And if you like what we talked about so far, please go to our website and apply to become a member of the Molecular Connectivity Working Group. Thank you.
Okay, thank you very much. This is the open part is, so to say, closed. So those with a badge, gray badge, yeah, thank you for the participation. Because the next parts will be closed, as indicated in the program. The rationale behind is very simple. We want to reach, I hope, that we will be that optimistic as Lucina, and we reach a certain minimal consensus in terminology and provide recommendations for the field, right? Therefore, we wanted to limit it to the members of the Molecular Connectivity Working Group, and there will be also no streaming. So just, yeah, to avoid any misunderstandings. Thank you very much. Colleagues with red badges, please stay in the room.